Welcome to an comprehensive exploration of one of the most fundamental and fascinating systems in the human body, the musculoskeletal system. This remarkable network of interconnected structures forms the very foundation of our physical existence, enabling everything from the delicate precision of threading a needle to the explosive power of an Olympic sprint. Understanding this system is not merely an academic exercise. It provides crucial insights into how our bodies move, adapt, and function in the complex environment we navigate daily. The musculoskeletal system represents a masterpiece of biological engineering, consisting of four primary components that work in seamless harmony. The skeletal system provides the rigid framework upon which everything else is built. Much like the steel girders of a skyscraper, our 206 bones serve multiple critical functions beyond simple structural support, acting as mineral reservoirs, sites of blood cell production, and protective shields for our most vital organs. The muscular system, comprised of over 600 individual muscles, generates the forces necessary for movement and maintains our posture against the constant pull of gravity. These muscles range from the massive gluteus maximus in our hips to the tiny stapedius muscle in our middle ear, each precisely designed for its specific function. The articular system, encompassing our joints and their associated structures, creates the pivotal points where movement occurs. Without joints, our skeleton would be nothing more than a rigid statue, incapable of the fluid motion that characterizes human movement. These joints are marvels of mechanical engineering, some designed for stability and others for mobility, with many achieving a remarkable balance between both demands. The connective tissue system weaves throughout the entire musculoskeletal framework, binding structures together while allowing for the flexibility and resilience necessary for dynamic function. This includes tendons that connect muscles to bones, ligaments that stabilize joints, and fascia that compartmentalizes and supports muscle groups. The integration of these four components creates a system of extraordinary sophistication and adaptability. Consider the simple act of reaching for a coffee cup on a table. This seemingly effortless movement requires the coordinated activation of dozens of muscles, the precise articulation of multiple joints, and the instantaneous processing of sensory feedback to adjust force and position. The bones provide stable platforms for muscle attachment, while the joints allow for the necessary range of motion. The muscles generate and control the forces, while connective tissues ensure that these forces are transmitted efficiently and that joints remain stable throughout the movement. The musculoskeletal system's importance extends far beyond enabling movement. It plays a crucial role in maintaining homeostasis through its involvement in mineral metabolism, particularly calcium and phosphate regulation. The bones serve as dynamic reservoirs for these essential minerals, releasing them into the bloodstream when needed and storing excess when abundant. The red bone marrow housed within certain bones is responsible for hematopoiesis, the production of blood cells that sustain life. Additionally, the muscular system contributes significantly to metabolic function, consuming glucose and generating heat through both voluntary contractions and involuntary metabolic processes. The system's remarkable ability to adapt and remodel in response to mechanical stress represents one of biology's most elegant examples of form-following function. Bones become denser and stronger when subjected to regular loading, while muscles hypertrophy when challenged with resistance. Conversely, both tissues atrophy when underused, demonstrating the principle of use it or lose it that governs much of human physiology. This adaptive capacity allows individuals to develop specialized capabilities, whether it's the dense bones of a weightlifter or the precise motor control of a surgeon. Understanding the musculoskeletal system also provides insight into the evolutionary history of human locomotion and manipulation. Our upright posture and bipedal gait have shaped every aspect of our musculoskeletal anatomy, from the shaped curve of our spine to the unique structure of our pelvis and the orientation of our hip joints. Our hands, freed from locomotor duties by bipedalism, have evolved into incredibly sophisticated tools capable of both power grips for climbing and precision grips for tool use and manipulation. The clinical significance of musculoskeletal knowledge cannot be overstated. Musculoskeletal disorders represent one of the leading causes of disability worldwide, affecting individuals across all age groups and activity levels. From the acute injuries sustained by athletes to the degenerative conditions that accompany aging, understanding the normal structure and function of this system is essential for recognizing pathology and implementing effective treatment strategies. Physical therapists, occupational therapists, physicians, and other healthcare professionals rely heavily on this knowledge to assess, diagnose, and treat a wide range of conditions. As we embark on this detailed exploration of the musculoskeletal system, we will examine each component in depth, understanding not only the individual structures but also their complex interactions and functional relationships. We will explore how bones develop and remodel, how muscles contract and generate force, how joints facilitate movement while maintaining stability, and how connective tissues provide the framework that binds it all together. This knowledge forms the foundation for understanding human movement, injury prevention, rehabilitation strategies, and the maintenance of musculoskeletal health throughout the lifespan. 2. Bone tissue fundamentals. Bone tissue represents one of the most remarkable materials found in nature, combining the strength of steel with the lightness of wood while maintaining the ability to repair and remodel itself throughout life. Unlike the static perception many people have of bones as merely dried calcium structures, living bone tissue is a dynamic, highly vascularized, and metabolically active tissue that continuously adapts to the mechanical demands placed upon it. This extraordinary material achieves its unique properties through a sophisticated hierarchical organization that spans from the molecular level to the gross anatomical structure. At the microscopic level, bone tissue exists in two primary forms, compact bone and spongy bone, also known as cortical and trabecular bone respectively. Compact bone forms the dense outer shell of all bones and comprises approximately 80% of the skeletal mass in the human body. This tissue is characterized by its tightly packed structure with minimal spaces, creating a material that can withstand tremendous compressive forces while providing excellent resistance to bending and torsional stresses. The fundamental structural unit of compact bone is the osteon, also called the Haversian system, which consists of concentric layers of bone matrix arranged around a central canal containing blood vessels and nerves. Each osteon typically measures between 200 to 400 micrometers in diameter and can extend several millimeters in length, running roughly parallel to the long axis of the bone. The central Haversian canal contains one or two capillaries and nerve fibers, providing the metabolic support necessary for the surrounding bone tissue. The concentric layers, called lamellae, consist of collagen fibers embedded in a calcified matrix, with each lamella containing collagen fibers oriented in a slightly different direction than adjacent layers. This alternating fiber orientation, similar to the construction of plywood, provides exceptional strength and resistance to fracture. Between the lamellae lie small spaces called lacunae, which house mature bone cells known as osteocytes. These cells extend numerous fine processes through tiny channels called canaliculi, creating an intricate network that allows for communication between cells and the transport of nutrients and waste products throughout the bone matrix. This cellular network is essential for maintaining bone health and coordinating the bone's response to mechanical stress and hormonal signals. Spongy bone, in contrast to compact bone, has a more porous structure, resembling a three-dimensional lattice or honeycomb. This organization, while appearing less organized than compact bone, is actually precisely arranged to provide maximum strength with minimal weight. The spaces between the bony trabeculae are filled with red bone marrow in certain locations, making spongy bone the primary site of blood cell formation in adults. The trabecular architecture is not random but follows the lines of mechanical stress, with trabeculae oriented along the directions of principal compressive and tensile forces. The bone matrix itself is a composite material consisting of approximately 35% organic components and 65% inorganic mineral deposits by dry weight. The organic component is primarily composed of type I collagen, a fibrous protein that provides tensile strength and flexibility to the bone. Collagen fibers are arranged in a specific pattern that can resist pulling forces while allowing for some degree of bending. Without this organic component, bones would be extremely brittle and would shatter easily under impact, much like a piece of chalk. The inorganic component consists primarily of calcium phosphate in the form of hydroxyapatite crystals, along with smaller amounts of calcium carbonate, magnesium, fluoride, and other minerals. These mineral deposits provide the compressive strength that allows bones to support body weight and resist crushing forces. The hydroxyapatite crystals are deposited along and between the collagen fibers in a highly organized manner, creating a composite material that is both strong and relatively lightweight. The blood supply to bone tissue is remarkably extensive, with bones receiving approximately 10% of the cardiac output, despite comprising only about 12% of body weight. This rich vascularization reflects the high metabolic activity of bone tissue and its role in mineral homeostasis. The periosteal vessels supply the outer layers of compact bone, while nutrient arteries penetrate the bone shaft through nutrient foramina to supply the inner regions of compact bone and the bone marrow. The metaphyseal and epiphyseal vessels supply the ends of long bones, creating a comprehensive network that ensures adequate oxygen and nutrient delivery to all regions of the bone. The periosteum, a dense, fibrous membrane covering the external surface of bones except at joint surfaces, plays a crucial role in bone nutrition, growth, and repair. The outer layer of the periosteum consists of dense, irregular connective tissue containing blood vessels and nerves, while the inner layer contains osteoprogenitor cells that can differentiate into osteoblasts when bone formation is required. This cellular reserve is particularly important during fracture healing and bone remodeling processes. The endosteum, a thin membrane lining the internal surfaces of bones, including the medullary cavity and the spaces within spongy bone, contains similar osteoprogenitor cells and provides another source of cells for bone remodeling and repair. The interface between the endosteum and bone tissue is where much of the bone remodeling activity occurs, with old bone being removed and new bone being deposited in response to mechanical stress and metabolic demands. Understanding these fundamental characteristics of bone tissue provides the foundation for comprehending how bones develop, grow, maintain themselves, and respond to injury throughout life. The dynamic nature of bone tissue, combined with its remarkable material properties, makes it uniquely suited to its multiple roles in the human body, from structural support to metabolic regulation. 3. Bone cells and bone remodeling. The living nature of bone tissue becomes most apparent when examining the specialized cells responsible for its formation, maintenance, and breakdown. These cells work in coordinated fashion throughout our lives, constantly remodeling our skeleton in response to mechanical stress, hormonal signals, and metabolic demands. This cellular activity ensures that our bones remain strong and healthy while adapting to changing functional requirements and repairing microscopic damage that occurs during normal daily activities. Osteoblasts represent the bone-building cells of the skeleton, responsible for synthesizing and secreting the organic matrix of bone tissue. These cells develop from mesenchymal stem cells through a complex differentiation process regulated by various growth factors and transcription factors. Once fully differentiated, osteoblasts are typically found arranged along bone surfaces where active bone formation is occurring. They appear as cuboidal or columnar cells with an abundant rough endoplasmic reticulum and large Golgi apparatus, reflecting their high protein synthesis activity. The primary function of osteoblasts is to produce and secrete type I collagen, the predominant protein in bone matrix, along with various non-collagenous proteins such as osteocalcin, osteonectin, and osteopontin. These proteins form the organic framework upon which mineral deposits will later be laid down. Osteoblasts also secrete alkaline phosphatase, an enzyme that plays a crucial role in the mineralization process by helping to precipitate calcium phosphate crystals within the organic matrix. The activity of alkaline phosphatase is so closely associated with bone formation that serum levels of this enzyme are commonly used as a clinical marker of bone formation activity. As osteoblasts continue to secrete matrix around themselves, some become trapped within the newly formed bone tissue and differentiate into osteocytes. This transformation involves a dramatic change in cell morphology and function, as the once active synthetic cells develop into a network of interconnected sensors that monitor the mechanical and chemical environment within the bone. The newly formed osteocytes establish communication with neighboring cells through gap junctions at the tips of their cellular processes, creating a three-dimensional network that can coordinate bone remodeling activities across large regions of bone tissue. Osteocytes represent the most abundant cell type in mature bone tissue, comprising over 90% of all bone cells. These cells occupy the lacunar canalicular system within the bone matrix, with their cell bodies residing in lacunae and their long, thin processes extending through canaliculi to connect with neighboring osteocytes and cells on bone surfaces. This cellular network functions as a sophisticated mechanosensory system, detecting changes in mechanical loading and initiating appropriate remodeling responses. The mechanosensory function of osteocytes relies on their ability to detect fluid flow within the canalicular system, caused by mechanical loading of the bone. When bone is subjected to stress, such as during walking or exercise, slight deformation occurs that causes interstitial fluid to flow through the canalicular network. Osteocytes detect this fluid flow through mechanosensitive ion channels and respond by releasing various signaling molecules that can either promote bone formation or bone resorption, depending on the type and magnitude of the mechanical stimulus. Osteoclasts represent the bone-resorbing cells responsible for breaking down and removing old or damaged bone tissue. Unlike osteoblasts and osteocytes, which derive from mesenchymal stem cells, osteoclasts originate from hematopoietic stem cells in the bone marrow and are related to macrophages and other immune system cells. These large, multinucleated cells can contain anywhere from 2 to 100 nuclei, reflecting their formation through the fusion of multiple mononuclear precursor cells. The bone resorption process carried out by osteoclasts is remarkably sophisticated and highly regulated. When activated, osteoclasts attach tightly to the bone surface through specialized adhesion structures called sealing zones, creating a sealed compartment between the cell and the bone matrix. Within this compartment, osteoclasts create an acidic environment by secreting hydrogen ions through proton pumps, lowering the pH to approximately 4.5. This acidic environment dissolves the mineral component of the bone matrix, primarily hydroxyapatite crystals. Simultaneously, osteoclasts secrete proteolytic enzymes, particularly cathepsin K, which breaks down the organic components of the bone matrix, primarily type I collagen. The combination of acid dissolution of minerals and enzymatic breakdown of organic components creates characteristic resorption pits called Hauschip's lacunae on the bone surface. The products of bone resorption, including calcium, phosphate, and collagen fragments, are then transported across the osteoclast and released into the circulation. The bone remodeling process represents the coordinated activity of these three cell types, working together to maintain skeletal integrity and respond to changing mechanical and metabolic demands. This process occurs continuously throughout life at numerous sites across the skeleton, with approximately 10% of the adult skeleton being remodeled each year. The remodeling process follows a predictable sequence beginning with activation, followed by resorption, reversal, formation, and finally termination. The activation phase involves the recruitment and activation of osteoclast precursors in response to various signals, including mechanical stress, hormonal influences, or microscopic damage to the bone matrix. During the resorption phase, Mature osteoclasts remove a packet of old bone tissue, creating a cavity that will subsequently be filled with new bone. The reversal phase represents a transition period during which osteoclastic activity ceases and osteoblastic activity begins. The formation phase involves the recruitment and activation of osteoblasts, which begin to synthesize new bone matrix to fill the cavity created during resorption. This phase typically takes several months to complete and results in the deposition of new bone tissue that is often stronger and better organized than the tissue it replaced. The termination phase occurs when the remodeling site returns to a quiescent state, with osteoblasts either undergoing apoptosis, remaining on the bone surface as lining cells, or becoming incorporated into the new bone matrix as osteocytes. This continuous remodeling process serves multiple important functions in maintaining skeletal health. It removes old, potentially weakened bone tissue before it can fail under normal loading conditions, replaces it with new, mechanically optimized tissue, and allows the skeleton to adapt its structure in response to changing mechanical demands. Additionally, the remodeling process plays a crucial role in calcium homeostasis, releasing stored calcium from the skeleton when blood calcium levels are low and depositing excess calcium when levels are high. The human skeleton contains 206 bones in the adult, each uniquely shaped and structured to fulfill specific mechanical and physiological functions. Understanding how bones are classified and organized provides crucial insight into their diverse roles and helps explain why different bones have evolved such distinct anatomical features. The classification of bones is based primarily on their shape and size, which directly relates to their functional requirements and the mechanical forces they must withstand during normal activities. Long bones represent perhaps the most recognizable category of skeletal elements and include the major bones of the appendicular skeleton, such as the femur, tibia, humerus, and radius. These bones are characterized by their elongated shaft, called the diaphysis, and their expanded ends, termed epiphyses. The diaphysis consists primarily of compact bone tissue arranged in a hollow cylindrical structure that provides maximum strength with minimal weight, similar to the engineering principle used in hollow steel beams. This design allows long bones to resist bending forces effectively, while keeping the overall mass of the skeleton manageable. The epiphyses of long bones are composed primarily of spongy bone tissue, covered by a thin layer of compact bone. This arrangement provides excellent shock absorption and distributes forces over a large surface area at joint interfaces. During development, the epiphyses are separated from the diaphysis by growth plates, areas of cartilage where longitudinal bone growth occurs. Once skeletal maturity is reached, these growth plates ossify and become the epiphyseal lines, permanent markers of where growth once occurred. Short bones are typically cube-shaped or irregular structures found primarily in the wrist and ankle regions. Examples include the carpal bones of the wrist and the tarsal bones of the foot. These bones consist mostly of spongy bone tissue covered by a thin layer of compact bone, making them excellent shock absorbers and load distributors. Their position in regions where multiple bones articulate allows them to transmit forces between different parts of the skeleton while providing the flexibility needed for complex movements. Flat bones, as their name suggests, are thin, flattened structures that typically serve protective functions or provide extensive surface areas for muscle attachment. The skull bones, ribs, sternum, and scapulae represent the primary examples of flat bones in the human body. These bones consist of two layers of compact bone tissue separated by a layer of spongy bone tissue, creating a structure similar to a sandwich. This design provides excellent protection for underlying organs while remaining relatively lightweight. The spongy bone tissue within flat bones often contains red bone marrow, making these bones important sites of blood cell production throughout life. The flat bones of the skull are particularly specialized, with the spongy bone layer called diploe containing numerous small veins that help regulate brain temperature and provide an additional route for venous drainage from the skull. Irregular bones encompass all bones that do not fit neatly into the other categories due to their complex, unique shapes. The vertebrae represent the most obvious examples of irregular bones, but this category also includes bones such as the hip bones, many facial bones, and the sacrum. These bones have evolved their complex shapes to fulfill multiple functions simultaneously, such as protecting organs, providing muscle attachment sites, and facilitating complex movements. Sesamoid bones represent a special category of bones that develop within tendons or joint capsules in response to mechanical stress. The patella, or kneecap, represents the largest and most consistent sesamoid bone in the human body, developing within the tendon of the quadriceps muscle group. Other sesamoid bones may develop in the hands and feet of some individuals, particularly in areas subject to repeated stress or friction. These bones serve to protect tendons from excessive wear and can also change the angle of tendon pull, potentially improving mechanical advantage for muscle contractions. Bone markings represent another crucial aspect of bone anatomy, providing information about the functional relationships between bones and the soft tissues that interact with them. These markings can be broadly categorized into processes, which represent projections or outgrowths from the bone surface, and depressions, which represent indentations or openings in the bone. Processes typically serve as attachment sites for muscles, tendons, or ligaments, while depressions often accommodate blood vessels, nerves, or articulating surfaces of other bones. Processes include structures such as tubercles, tuberosities, trochanters, spines, and crests, each representing different sizes and shapes of projections from the bone surface. Tuberosities and trochanters represent large, rough projections that serve as attachment sites for major muscle groups, while tubercles are smaller, rounded projections for individual muscles or ligaments. Spines and crests represent sharp, ridge-like projections that often serve as attachment sites for sheets of connective tissue or multiple muscle groups. Depressions and openings include structures such as fossae, grooves, foramina, canals, and fissures. Fossae represent shallow depressions that often accommodate muscle bellies or other soft tissue structures. Grooves provide pathways for blood vessels or nerves along the bone surface. Foramina represent circular openings through bones that allow passage of nerves and blood vessels, while canals represent longer tunnels through bone tissue. Fissures represent narrow, slit-like openings that also accommodate neural or vascular structures. The anatomical organization of individual bones reflects their developmental history and functional requirements. During development, most bones form through the process of endochondral ossification, beginning as cartilage models that are gradually replaced by bone tissue. This process leaves characteristic features such as the epiphyseal lines mentioned earlier, as well as various surface features that reflect the attachment points of muscles and ligaments during development. Understanding bone anatomy also requires appreciation of the relationship between form and function that governs skeletal architecture. Bones subjected to compressive forces, such as the vertebrae and the bones of the lower limb, tend to have thick, robust construction, with extensive spongy bone tissue to distribute loads. Bones subjected primarily to tensile or bending forces, such as the ribs and many bones of the upper limb, tend to have more slender construction with emphasis on compact bone tissue for strength. The surface features of bones also reflect their mechanical environment, with roughened areas indicating sites of strong muscle or ligament attachment, and smooth areas representing regions where bones articulate with each other, or where soft tissues glide over bone surfaces. This intimate relationship between structure and function makes bone anatomy a particularly fascinating subject that provides insights into both human evolution and individual adaptation to mechanical demands. 5. The axial skeleton. Skull. The human skull represents one of the most complex and fascinating structures in the entire skeletal system, serving simultaneously as a protective vault for the brain, a framework for the sensory organs, and the foundation for the digestive and respiratory systems. comprising 22 individual bones that fuse during development to create a remarkably strong yet relatively lightweight structure. The skull demonstrates extraordinary engineering principles that have evolved over millions of years to meet the unique demands of human anatomy and behavior. The cranial portion of the skull, consisting of 8 bones, forms the protective enclosure for the brain and provides attachment sites for many of the muscles involved in head and neck movement. The frontal bone forms the forehead and the anterior portion of the skull roof, containing the frontal sinuses that help reduce the weight of the skull while maintaining strength. The frontal bone also forms the superior portions of the eye sockets, called the orbits, and contains the supraorbital foramina that allow passage of nerves and blood vessels to the forehead region. The parietal bones, typically numbering 2, form the majority of the skull roof and the superior portions of the skull sides. These bones are separated by the sagittal suture, one of several fibrous joints that allow for brain growth during infancy and childhood, while eventually fusing to create a solid protective vault. The parietal bones demonstrate the classic construction of flat bones, with outer and inner layers of compact bone separated by spongy bone tissue containing diploic veins that help regulate brain temperature. The temporal bones, located on either side of the skull, house the organs of hearing and balance while providing attachment sites for powerful jaw muscles. Each temporal bone contains the external auditory meatus, the opening that leads to the middle and inner ear structures. The temporal bones also feature the mastoid processes, large projections behind the ears that serve as attachment sites for neck muscles and contain air cells that communicate with the middle ear. The temporal bones articulate with the mandible through the temporomandibular joints, the only movable joints in the skull. The occipital bone forms the posterior and inferior portions of the skull, containing the foramen magnum, the large opening through which the brainstem connects with the spinal cord. The occipital bone features prominent external and internal projections that serve as attachment sites for neck muscles and provide anchoring points for membranes that separate different regions of the brain. The occipital condyles, located on either side of the foramen magnum, articulate with the first cervical vertebra to form the joint that allows nodding movements of the head. The sphenoid bone, often called the keystone of the skull, occupies a central position and articulates with virtually every other skull bone. This complex bone forms part of the skull base and contains the sella turcica, a depression that houses the pituitary gland. The sphenoid also contains the sphenoid sinuses and features numerous foramina that allow passage of important nerves and blood vessels, including the optic canals for the optic nerves and the superior orbital fissures for nerves controlling eye movement. The ethmoid bone, located between the nasal cavity and the brain, forms part of the nasal septum and the lateral walls of the nasal cavity. This delicate bone contains the cribriform plate, a perforated structure that allows olfactory nerves to pass from the nasal cavity to the brain, enabling our sense of smell. The ethmoid also contains extensive air cells that help humidify and warm inspired air while reducing the weight of the skull. The facial skeleton, comprising 14 bones, provides the structural framework for the face while housing and protecting the organs of vision, smell, and taste. The maxillae, or upper jaw bones, form much of the middle face and contain the maxillary sinuses, the largest of the paranasal sinuses. The maxillae hold the upper teeth and form most of the hard palate, the bony roof of the mouth that separates the oral and nasal cavities. These bones also contribute to the formation of the orbits and the lateral walls of the nasal cavity. The mandible, or lower jaw bone, represents the largest and strongest facial bone, and the only movable bone of the skull. This horseshoe-shaped bone holds the lower teeth and provides attachment sites for the powerful muscles of mastication. The mandible features 2 vertical portions called rami that articulate with the temporal bones, and a horizontal portion called the body that forms the chin and supports the lower teeth. The mandible contains the mandibular foramen on each side, allowing passage of nerves and blood vessels that supply sensation to the lower teeth and lip. The nasal bones form the bridge of the nose and provide attachment for the nasal cartilages that shape the external nose. Despite their small size, these bones play an important role in facial appearance and are frequently involved in facial fractures. The lacrimal bones, the smallest bones of the face, contain the lacrimal fossae that house the lacrimal sacs, part of the tear drainage system that keeps our eyes moist and clean. The palatine bones contribute to the posterior portion of the hard palate and the lateral walls of the nasal cavity. These L-shaped bones help separate the nasal and oral cavities while providing additional surface area for the nasal mucosa that warms and humidifies inspired air. The zygomatic bones form the cheekbones and contribute to the lateral walls of the orbits, providing attachment sites for muscles of facial expression and helping to distribute forces generated during chewing. The inferior nasal conchae extend from the lateral walls of the nasal cavity and help create turbulent airflow that increases the contact between inspired air and the nasal mucosa. This turbulence enhances the warming, humidification, and filtration of inspired air before it reaches the lungs. The vomer forms the posterior portion of the nasal septum, working together with the ethmoid bone to separate the left and right nasal cavities. Sutures represent the fibrous joints that connect most skull bones and demonstrate different patterns depending on the mechanical forces they must resist. The coronal suture connects the frontal and parietal bones. The sagittal suture connects the two parietal bones, and the lambdoid suture connects the parietal and occipital bones. These sutures remain flexible during infancy and childhood, allowing for brain growth and passage through the birth canal, but gradually ossify during adulthood to create a solid, protective vault. The skull also contains numerous foramina, canals, and fissures that allow passage of nerves, blood vessels, and other structures between the interior and exterior of the skull. Major openings include the optic canals for the optic nerves, the superior and inferior orbital fissures for nerves controlling eye movement and facial sensation, the foramen rotundum, ovale, and spinosum for branches of the trigeminal nerve, and the internal auditory meatusses for the nerves controlling hearing and balance. Understanding skull anatomy provides crucial insights into human evolution, development, and clinical medicine, as this complex structure reflects our species' unique adaptations for large brain size, binocular vision, and complex social communication through facial expression and speech. 6. The axial, skeleton, vertebral column. The vertebral column represents one of the most remarkable achievements in vertebrate evolution, providing a flexible yet strong axis that supports the weight of the head and trunk, while protecting the delicate spinal cord and allowing for a wide range of movements. Consisting of 33 individual vertebrae during fetal development, which later fused to form 26 separate bones in the adult, the vertebral column demonstrates how structure and function are intimately related in biological systems. The column's characteristic shaped curvature when viewed from the side provides optimal weight distribution and shock absorption, while maintaining balance during upright posture. The cervical region of the vertebral column consists of 7 vertebrae, labeled cone through C7, which support the weight of the head while allowing for the extensive range of motion necessary for visual tracking, communication, and environmental awareness. The first 2 cervical vertebrae, the atlas and axis, are highly specialized to accommodate the unique mechanical demands of supporting and moving the head. The atlas, or cone, lacks a vertebral body and instead consists of anterior and posterior arches connected by lateral masses. This ring-like structure articulates with the occipital condyles of the skull, forming the atlanto-occipital joint that allows for nodding movements of the head, such as those used to indicate yes. The axis, or C2, features a unique upward projection called the odontoid process, or dens, which extends through the anterior arch of the atlas and serves as a pivot point for rotational movements of the head. This arrangement creates the atlanto-axial joint, which allows for approximately 50% of the total rotational movement of the head and neck, such as the motion used to indicate no. The remaining 5 cervical vertebrae, C3 through C7, share certain common characteristics while showing progressive changes that reflect the increasing load-bearing requirements as one moves down the column. Typical cervical vertebrae are characterized by their small, delicate construction, reflecting the relatively light loads they must support. Their vertebral bodies are small and wider from side to side than from front to back, while their spinous processes are typically bifid, or split into two parts. One of the most distinctive features of cervical vertebrae is the presence of transverse foramina in their transverse processes, which accommodate the vertebral arteries that supply blood to the brain. This vascular arrangement is unique to the cervical region and reflects the critical importance of maintaining adequate blood flow to the brain during neck movements. The seventh cervical vertebra, known as the vertebra prominens, serves as a transition between the cervical and thoracic regions. Its spinous process is particularly prominent and can be easily palpated at the base of the neck, serving as an important anatomical landmark for clinicians and anatomists. The C7 vertebra also typically lacks the bifid spinous process characteristic of other cervical vertebrae and may not have transverse foramina, reflecting its transitional nature. The thoracic region consists of 12 vertebrae, toned through 12, which articulate with the ribs to form the thoracic cage. These vertebrae demonstrate progressive increases in size and strength as they support increasing amounts of body weight moving down the column. The most distinctive feature of thoracic vertebrae is the presence of costal facets, special articular surfaces that accommodate the heads and tubercles of the ribs. The upper thoracic vertebrae typically have full costal facets on their bodies and transverse processes, while the lower thoracic vertebrae show progressive reduction in these features as the rib attachments become less robust. The bodies of thoracic vertebrae are intermediate in size between the smaller cervical vertebrae and the larger lumbar vertebrae, reflecting their intermediate position and load-bearing requirements. Their spinous processes are long and slender, angled sharply downward in an overlapping pattern that provides additional stability to the thoracic region, while limiting extension movements that might compromise the protection of vital organs within the thoracic cavity. The thoracic region of the vertebral column demonstrates a kyphotic curvature, meaning it curves backward when viewed from the side. This curvature is present from birth and represents the primary curvature of the vertebral column, retained from the C-shaped curvature present during fetal development. The thoracic kyphosis helps position the rib cage optimally for respiratory function while contributing to the overall balance and shock absorption capabilities of the vertebral column. The lumbar region consists of 5 large, robust vertebrae, lown through L5, which bear the greatest loads in the vertebral column and consequently show the most massive construction. Lumbar vertebrae have large, kidney-shaped vertebral bodies and thick, blunt spinous processes that project directly posterior rather than at the downward angle seen in thoracic vertebrae. The transverse processes of lumbar vertebrae are also well developed, providing extensive attachment sites for the powerful muscles that control trunk movement and maintain posture. The lumbar region demonstrates a lordotic curvature, meaning it curves forward when viewed from the side. This curvature develops as children begin to walk and represents a secondary curvature that helps position the body's center of gravity over the lower limbs during upright posture. The lumbar lordosis is maintained primarily by the wedge shape of the intervertebral discs in this region and the orientation of the facet joints, which are arranged to resist forward sliding of the vertebrae while allowing flexion and extension movements. The sacral region consists of 5 vertebrae, sone through S5, which fuse during late adolescence and early adulthood to form the sacrum, a single triangular bone that forms the posterior wall of the pelvis. The sacrum articulates with the hip bones on either side through the sacroiliac joints, creating a stable foundation for the transfer of weight from the vertebral column to the lower limbs. The anterior surface of the sacrum is smooth and concave, forming part of the posterior wall of the pelvic cavity, while the posterior surface is rough and convex, providing attachment sites for numerous muscles and ligaments. The sacrum contains 4 pairs of sacral foramina on both its anterior and posterior surfaces, which allow passage of the sacral nerves that supply the pelvis and lower limbs. The superior portion of the sacrum, formed primarily by the first sacral vertebra, articulates with the 5th lumbar vertebra at the lumbosacral joint, which is subject to considerable mechanical stress due to the change in orientation between the mobile lumbar spine and the relatively fixed sacrum. The coccygeal region consists of 3 to 5 small, rudimentary vertebrae that fuse to form the coccyx or tailbone. This structure represents the evolutionary remnant of a tail and serves as an attachment site for several muscles and ligaments associated with the pelvic floor. Despite its small size and vestigial nature, the coccyx plays an important role in supporting body weight during sitting and provides crucial attachment points for muscles involved in bowel and bladder control. The intervertebral discs represent crucial components of the vertebral column that are not technically part of the skeletal system but are essential for understanding spinal function. These fibrocartilaginous structures are located between adjacent vertebral bodies from the 2nd cervical vertebra to the sacrum, comprising approximately 25% of the total height of the vertebral column. Each disc consists of a tough outer ring called the annulus fibrosus and a gel-like center called the nucleus pulposus, which together provide shock absorption and allow for movement between vertebrae while maintaining stability. The curvatures of the vertebral column play crucial roles in maintaining balance, distributing weight, and absorbing shock during locomotion. The primary curvatures present at birth include the thoracic kyphosis and the sacral kyphosis, which retain the C-shaped curvature of the fetal spine. The secondary curvatures, which develop as children learn to hold up their heads and walk, include the cervical lordosis and the lumbar lordosis. These curvatures create the characteristic shape of the adult vertebral column, which provides optimal mechanical advantage for upright posture while maintaining flexibility for movement. 7. The axial skeleton, thoracic cage. The thoracic cage represents a remarkable example of biological engineering, forming a protective yet flexible enclosure for the vital organs of circulation and respiration, while providing essential mechanical support for breathing and upper limb function. Consisting of the sternum, 12 pairs of ribs, and their associated costal cartilages, along with the 12 thoracic vertebrae posteriorly, the thoracic cage demonstrates how skeletal structures can simultaneously provide protection, support, and mobility. This complex arrangement allows for the continuous expansion and contraction necessary for breathing while maintaining protection for the heart, lungs, and major blood vessels. The sternum, or breastbone, forms the central anterior component of the thoracic cage and consists of 3 fused segments that demonstrate the developmental history of this important structure. The manubrium represents the superior portion of the sternum and articulates with the clavicles at the sternoclavicular joints, the only direct skeletal connection between the upper limbs and the axial skeleton. The manubrium also provides attachment sites for the first ribs and the upper portion of the second ribs, creating stable anchor points for the superior aspect of the thoracic cage. The body of the sternum forms the longest portion of this bone and provides attachment sites for the costal cartilages of ribs 2 through 7. The lateral borders of the sternal body feature distinct notches that accommodate these cartilages, creating secure but flexible connections that allow for respiratory movements while maintaining thoracic cage integrity. The sternal body also serves as an important attachment site for the pectoralis major muscles, which are crucial for upper limb function and respiratory assistance during forced breathing. The xiphoid process represents the small, inferior portion of the sternum and demonstrates considerable individual variation in size and shape. This structure provides attachment sites for several muscles of the anterior abdominal wall and serves as an important landmark for cardiopulmonary resuscitation and other clinical procedures. The xiphoid process typically remains cartilaginous until middle age, when it gradually ossifies and fuses with the sternal body. The 12 pairs of ribs demonstrate a fascinating combination of similarity and variation that reflects their different mechanical roles within the thoracic cage. All ribs share certain basic structural features, including a head that articulates with vertebral bodies, a neck that provides leverage for respiratory movements, a tubercle that articulates with transverse processes of thoracic vertebrae, and a shaft that curves around the thoracic cavity to provide protection and support. However, the specific characteristics of individual ribs reflect their position within the thoracic cage and their particular functional requirements. The first 7 pairs of ribs, known as true ribs or vertebrosternal ribs, attach directly to the sternum through their individual costal cartilages. These ribs demonstrate the most robust construction and provide the primary structural framework for the upper portion of the thoracic cage. The first rib is particularly specialized, being shorter and more curved than the others, with distinct grooves on its superior surface that accommodate the subclavian vessels and provide attachment sites for respiratory muscles. The 8th, 9th, and 10th ribs, known as false ribs or vertebrochondral ribs, do not attach directly to the sternum, but instead connect to the costal cartilage of the 7th rib through their individual costal cartilages. This arrangement allows for greater flexibility in the lower portion of the thoracic cage, while maintaining structural integrity and protection for the lower portions of the lungs and upper abdominal organs. The 11th and 12th ribs, known as floating ribs or vertebral ribs, have no anterior attachment and end freely in the posterior abdominal wall musculature. These ribs are shorter than the others and provide protection for the kidneys and other posterior abdominal structures, while allowing for maximum flexibility during trunk movements and deep breathing. The costal cartilages represent crucial components of the thoracic cage that are often overlooked but play essential roles in respiratory function and thoracic cage mechanics. These hyaline cartilage structures provide flexible connections between the ribs and sternum or between lower ribs and adjacent costal cartilages. The flexibility of costal cartilages is essential for the expansion and contraction of the thoracic cage during breathing, as the relatively rigid bony ribs alone would not permit the volume changes necessary for effective ventilation. The shape and orientation of the ribs create the characteristic barrel-like appearance of the thoracic cage and establish the mechanical basis for respiratory movements. When viewed from the side, each rib slopes downward from its vertebral attachment to its sternal attachment, creating what is known as the bucket handle mechanism of breathing. During inspiration, the ribs rotate upward around an axis running from the head to the tubercle of each rib, increasing both the anteroposterior and lateral dimensions of the thoracic cavity. The intercostal spaces between adjacent ribs house the intercostal muscles, which play crucial roles in breathing and provide additional structural support for the thoracic cage. The external intercostal muscles, with fibers running downward and forward, assist in inspiration by elevating the ribs, while the internal intercostal muscles, with fibers running downward and backward, assist in forced expiration by depressing the ribs. These muscles, along with the diaphragm, create the pressure changes necessary for air movement into and out of the lungs. The thoracic cage also provides important attachment sites for numerous muscles involved in upper limb function, breathing, and trunk stability. The pectoralis major and minor muscles attach to the anterior thoracic cage and play crucial roles in shoulder and arm movements. The serratus anterior muscle attaches along the lateral aspects of the upper ribs and is essential for scapular movement and shoulder stability. Posteriorly, numerous muscles of the back attach to the ribs and thoracic vertebrae, contributing to posture and trunk movement. The protective function of the thoracic cage cannot be overstated, as it houses and shields the heart, lungs, and major blood vessels from external trauma, while maintaining the pressure differentials necessary for cardiac and respiratory function. The cage-like construction distributes impact forces over a wide area, while the flexibility provided by costal cartilages helps absorb energy from impacts that might otherwise cause serious injury to internal organs. The thoracic cage also plays important roles in venous return and lymphatic drainage through the rhythmic pressure changes associated with breathing. The respiratory pump mechanism assists in moving blood through the venous system back to the heart and helps propel lymph through the lymphatic system, contributing to immune function and fluid balance throughout the body. Understanding the anatomy and function of the thoracic cage is essential for comprehending not only respiratory mechanics but also the complex interactions between the skeletal, muscular, and cardiovascular systems that maintain homeostasis and enable the wide range of human activities that depend on effective oxygen delivery and carbon dioxide removal. 8. The appendicular skeleton. Shoulder girdle. The shoulder girdle represents one of the most mobile and functionally sophisticated regions of the human skeleton, providing the essential foundation for upper limb function, while demonstrating remarkable adaptations for the wide range of movements required for manipulation, tool use, and complex motor tasks. Consisting of the clavicle and scapula on each side of the body, the shoulder girdle creates a movable platform that positions the upper limb optimally for reaching, grasping, and manipulating objects throughout a large volume of space around the body. This mobility comes at the cost of inherent instability, requiring complex muscular coordination to maintain proper alignment and function. The clavicle, or collarbone, serves as the primary structural link between the upper limb and the axial skeleton, functioning essentially as a strut that holds the shoulder away from the chest wall and maintains optimal positioning for upper limb function. This shaped bone is unique in being the only long bone that lies horizontally in the body, and the only skeletal connection between the upper limb and the trunk, making it crucial for transmitting forces between these regions during activities involving the arms and hands. The medial end of the clavicle, enlarged and triangular in shape, articulates with the manubrium of the sternum at the sternoclavicular joint, the only true synovial joint connecting the upper limb to the axial skeleton. This joint is remarkably mobile, allowing the clavicle to move in multiple directions during shoulder and arm movements. The sternoclavicular joint is supported by strong ligaments that help prevent dislocation while allowing for the extensive range of motion necessary for optimal shoulder function. The lateral end of the clavicle is flattened and articulates with the acromion process of the scapula at the acromioclavicular joint. This joint, while less mobile than the sternoclavicular joint, still allows for important adjustments in scapular position during arm movements. The acromioclavicular joint is frequently subject to injury, particularly in contact sports and activities involving overhead arm movements, due to its superficial location and the forces transmitted through it during upper limb activities. The shaft of the clavicle demonstrates the characteristic S-curve that gives this bone its distinctive appearance and functional properties. The medial 2/3 of the shaft curves anteriorly, while the lateral 1/3 curves posteriorly, creating a configuration that provides optimal mechanical advantage for muscle attachments while maintaining adequate space for underlying neurovascular structures. The clavicle also serves as an attachment site for several important muscles, including the pectoralis major, deltoid, and trapezius muscles. The scapula, or shoulder blade, represents one of the most morphologically complex bones in the human body, featuring multiple processes, fossae, and borders that reflect its numerous muscular attachments and functional requirements. This flat, triangular bone lies against the posterior aspect of the thoracic cage, held in position entirely by muscular attachments rather than direct skeletal connections. This arrangement allows the scapula to glide freely over the thoracic wall, contributing significantly to the overall mobility of the shoulder complex. The body of the scapula forms the main triangular portion of the bone and is divided into anterior and posterior surfaces by the spine of the scapula, a prominent ridge that runs diagonally across the posterior surface. The supraspinous fossa, located above the spine, provides attachment for the supraspinatus muscle, while the larger infraspinous fossa, located below the spine, houses the infraspinatus muscle. These muscles, along with the subscapularis muscle that occupies the anterior subscapular fossa, form part of the rotator cuff mechanism that stabilizes the shoulder joint. The spine of the scapula extends laterally to form the acromion process, a flattened projection that articulates with the clavicle and provides attachment sites for the deltoid and trapezius muscles. The acromion also forms part of the roof of the shoulder joint, working together with the coracoacromial ligament to create a protective arch over the head of the humerus. Variations in acromion shape and orientation can influence shoulder function and contribute to impingement syndromes that affect overhead arm movements. The coracoid process represents another important projection from the scapula, extending anteriorly and slightly laterally from the superior border of the scapular body. This hook-like process provides attachment sites for several muscles, including the pectoralis minor, coracobrachialis, and the short head of the biceps brachii, as well as important ligaments that help stabilize the shoulder joint. The coracoid process also serves as a landmark for various clinical procedures and anatomical relationships in the shoulder region. The glenoid cavity, located at the lateral angle of the scapula, forms the socket portion of the shoulder joint and articulates with the head of the humerus. This shallow, pear-shaped depression is surrounded by the glenoid labrum, a rim of fibrocartilage that slightly deepens the socket and helps stabilize the joint. The shallow nature of the glenoid cavity contributes to the remarkable mobility of the shoulder joint, while simultaneously making it the most frequently dislocated major joint in the body. The borders and angles of the scapula provide additional attachment sites for muscles that control scapular position and movement. The medial border provides attachment for the rhomboid muscles and the middle portion of the trapezius muscle, while the lateral border serves as an attachment site for the teres major and teres minor muscles. The superior border contains the scapular notch, through which the suprascapular nerve passes to innervate muscles on the posterior aspect of the scapula. The movement of the shoulder girdle involves complex coordination between the sternoclavicular, acromioclavicular, and scapulothoracic joints, creating what is known as the shoulder girdle complex. During arm elevation, the scapula rotates upward, while the clavicle elevates and rotates posteriorly, maintaining optimal alignment of the glenoid cavity with the humeral head throughout the range of motion. This scapulohumeral rhythm is essential for normal shoulder function and helps prevent impingement of soft tissues during overhead activities. The muscular control of the shoulder girdle involves numerous muscles that can be broadly categorized based on their effects on scapular position. The trapezius muscle, with its upper, middle, and lower portions, provides elevation, retraction, and upward rotation of the scapula respectively. The serratus anterior muscle produces protraction and upward rotation, while the rhomboids and levator scapulae produce retraction, downward rotation, and elevation. The coordinated action of these muscles is essential for maintaining proper scapular positioning and enabling optimal upper limb function. The shoulder girdle's adaptation for mobility rather than stability reflects the evolutionary history of human upper limb function, with natural selection favoring configurations that maximize reaching ability and manipulation skills over pure strength or stability. This specialization has made humans exceptionally capable of complex manual tasks, but has also created a region that is susceptible to injury and dysfunction when the delicate balance between mobility and stability is disrupted. 9. The appendicular skeleton, upper limb bones. The bones of the upper limb represent a masterpiece of evolutionary engineering, perfectly adapted for the complex tasks of reaching, grasping, and manipulating objects with extraordinary precision and strength. From the powerful humerus that provides leverage for major movements to the delicate bones of the fingers that enable fine motor control, each bone in the upper limb demonstrates specific adaptations that reflect its functional role in the kinetic chain of arm and hand movement. The progressive decrease in bone size from proximal to distal combined with the increasing complexity of joint arrangements, creates a system capable of both powerful movements and delicate manipulations. The humerus, the longest and largest bone of the upper limb, extends from the shoulder to the elbow and serves as the primary lever arm for upper limb movements. The proximal end of the humerus features a smooth, rounded head that articulates with the glenoid cavity of the scapula to form the shoulder joint. This ball and socket arrangement provides the greatest range of motion of any joint in the body, allowing for movement in all three planes of motion. The head of the humerus is oriented at approximately a 130-degree angle to the shaft, positioning it optimally for articulation with the scapula, while providing adequate space for the extensive muscle attachments around the shoulder. The anatomical neck of the humerus represents a slight constriction immediately below the humeral head, marking the boundary between the articular surface and the main body of the bone. The surgical neck, located below the tubercles, represents the most common site of humeral fractures and derives its name from its clinical significance, rather than any distinct anatomical feature. This region is particularly vulnerable to fracture due to the transition from the expanded proximal end to the narrower shaft, creating a stress concentration point. The greater and lesser tubercles of the humerus provide attachment sites for the four rotator cuff muscles that are crucial for shoulder stability and function. The greater tubercle, located laterally, serves as the insertion point for the supraspinatus, infraspinatus, and teres minor muscles, while the lesser tubercle, located anteriorly, provides attachment for the subscapularis muscle. Between these tubercles lies the intertubercular groove, also known as the bicipital groove, which accommodates the tendon of the long head of the biceps brachii muscle. The shaft of the humerus demonstrates the characteristic features of a typical long bone, with a hollow medullary cavity surrounded by compact bone tissue that provides strength while minimizing weight. The mid-shaft region features the deltoid tuberosity, a roughened area that serves as the insertion point for the powerful deltoid muscle responsible for shoulder abduction. The shaft also contains the spiral groove, also called the radial groove, which accommodates the radial nerve and deep brachial artery as they course around the posterior aspect of the humerus. The distal end of the humerus expands to form the elbow joint with the radius and ulna, creating a complex articulation that allows for both flexion-extension movements and pronation-supination of the forearm. The capitulum, a rounded projection on the lateral side, articulates with the head of the radius, while the trochlea, a pulley-shaped structure on the medial side, articulates with the trochlear notch of the ulna. This arrangement creates a stable hinge joint for flexion and extension, while allowing the radius to rotate during pronation and supination. The medial and lateral epicondyles of the humerus provide attachment sites for numerous muscles that control forearm and wrist movements. The medial epicondyle, larger and more prominent, serves as the common origin for the flexor muscles of the forearm, while the lateral epicondyle provides attachment for the extensor muscles. These bony prominences are commonly involved in overuse injuries such as golfer's elbow and tennis elbow, which result from repetitive stress on the attached muscle groups. The ulna, the medial bone of the forearm, serves as the primary stabilizing element of the elbow joint and provides a stable platform for forearm rotation. The proximal end of the ulna features the prominent olecranon process, which forms the point of the elbow and provides attachment for the triceps brachii muscle. The trochlear notch, a deep C-shaped depression, articulates with the trochlea of the humerus to create the primary hinge mechanism of the elbow joint. The coronoid process of the ulna projects anteriorly from the proximal end and provides attachment for the brachialis muscle, while helping to prevent hyperextension of the elbow. The radial notch, located on the lateral aspect of the proximal ulna, accommodates the head of the radius and allows for rotation of the radius around the ulna during pronation and supination movements. The shaft of the ulna demonstrates a characteristic triangular cross-section with 3 borders and 3 surfaces, providing attachment sites for numerous muscles of the forearm while maintaining structural integrity. The interosseous border of the ulna provides attachment for the interosseous membrane that connects the ulna and radius along their length, helping to distribute forces between the two bones and providing additional attachment sites for forearm muscles. The radius, the lateral bone of the forearm, serves as the primary weight-bearing bone at the wrist and plays a crucial role in forearm rotation. The proximal end of the radius features a disc-shaped head that articulates with both the capitulum of the humerus and the radial notch of the ulna. This unique arrangement allows the radius to both participate in elbow flexion extension and rotate around the ulna during pronation supination. The radial tuberosity, located just below the neck of the radius, provides attachment for the biceps brachii muscle, which is responsible for both elbow flexion and forearm supination. The shaft of the radius demonstrates progressive enlargement from proximal to distal, reflecting its increasing importance as a weight-bearing structure approaching the wrist joint. The distal end of the radius expands significantly to form the primary articulation with the carpal bones of the wrist. The styloid process of the radius extends distally on the lateral side, providing attachment for the brachioradialis muscle and serving as an important anatomical landmark. The distal radius contains the scaphoid and lunate fossae, which accommodate the two primary carpal bones that transmit forces from the hand to the forearm. The bones of the hand represent perhaps the most complex and functionally sophisticated arrangement of skeletal elements in the human body, enabling the remarkable dexterity and precision that characterizes human manual capabilities. The hand contains 27 individual bones arranged in 3 distinct groups: the carpal bones of the wrist, the metacarpal bones of the palm, and the phalanges of the fingers. The 8 carpal bones are arranged in 2 rows and create a complex joint system that allows for the multi-directional movements of the wrist while providing stability for hand function. The proximal row, from lateral to medial, consists of the scaphoid, lunate, triquetrum, and pisiform bones, while the distal row contains the trapezium, trapezoid, capitate, and hamate bones. These bones are bound together by numerous ligaments and form several distinct joint spaces that contribute to wrist mobility and stability. The 5 metacarpal bones form the structural framework of the palm and are numbered from 1 to 5, beginning with the thumb. Each metacarpal consists of a base that articulates with carpal bones, a shaft that provides structural support, and a head that articulates with the proximal phalanges. The metacarpals demonstrate progressive changes in size and mobility, with the first metacarpal being the most mobile to accommodate thumb opposition, while the second and third metacarpals are the most stable to provide a solid foundation for grip strength. The phalanges, or finger bones, total 14 in number, with 3 in each finger and 2 in the thumb. The different lengths and proportions of these bones, combined with the complex arrangement of joints between them, create the precise control and adaptability that makes human hand function so remarkable. The distal phalanges feature expanded tips that provide support for the fingertips and nail beds, crucial structures for tactile sensation and precise manipulation of small objects. The pelvic girdle stands as one of the most structurally robust and functionally critical regions of the human skeleton, serving simultaneously as a stable foundation for bipedal locomotion, a protective enclosure for pelvic organs, and a crucial link in the transmission of forces between the vertebral column and the lower limbs. Unlike the shoulder girdle, which prioritizes mobility over stability, the pelvic girdle demonstrates the opposite evolutionary strategy, sacrificing some range of motion to achieve the structural integrity necessary for supporting body weight and enabling efficient upright walking. This fundamental difference in design philosophy reflects the distinct functional demands placed on the upper and lower limb girdles in human anatomy. Each hip bone, also known as the coxal bone or innominate bone, represents a complex fusion of 3 originally separate bones that unite during development to create a single, robust structure capable of withstanding the tremendous forces generated during locomotion and weight-bearing activities. The ilium, ischium, and pubis begin as separate ossification centers during fetal development but gradually fuse throughout childhood and adolescence, with complete fusion typically achieved by the early 20s. The point where these three bones meet is marked by the acetabulum, the deep socket that accommodates the head of the femur to form the hip joint. The ilium forms the superior and largest portion of the hip bone, characterized by its fan-shaped structure that provides extensive surface area for muscle attachments, while creating the prominent hip region that gives the human pelvis its distinctive appearance. The iliac crest represents the superior border of the ilium and can be easily palpated along its entire length, serving as an important anatomical landmark for clinical examinations and anatomical orientation. This curved ridge provides attachment sites for numerous muscles involved in trunk stability and lower limb movement, including the external oblique, internal oblique, transversus abdominis, and tensor fasciae latae muscles. The anterior superior iliac spine marks the anterior termination of the iliac crest and serves as the origin for the sartorius muscle, the longest muscle in the human body. This bony prominence is easily palpable in most individuals and represents a crucial landmark for anatomical measurements and clinical assessments. The anterior inferior iliac spine, located below the anterior superior spine, provides attachment for the rectus femoris muscle, one of the four muscles comprising the quadriceps femoris group. The posterior aspect of the ilium features the posterior superior and posterior inferior iliac spines, which provide attachment sites for various muscles and ligaments involved in sacroiliac joint stability and hip function. The auricular surface of the ilium articulates with the corresponding surface of the sacrum to form the sacroiliac joint, a crucial articulation that transfers weight from the vertebral column to the lower limbs, while allowing for slight movements that help absorb shock during locomotion. The ischium forms the posterior and inferior portion of the hip bone and demonstrates the most robust construction of the three component bones, reflecting its role in supporting body weight during sitting and its contribution to the stability of the hip joint. The body of the ischium contributes to the acetabulum and provides attachment sites for powerful muscles involved in hip extension and adduction. The ischial tuberosity represents a large, roughened projection that serves as the attachment site for the hamstring muscles and supports body weight during sitting. The ischial spine, located on the posterior border of the ischium, provides attachment for the sacrospinous ligament and serves as an important landmark that divides the greater sciatic notch from the lesser sciatic notch. These notches, converted into foramina by the sacrotuberous and sacrospinous ligaments, provide passageways for nerves and blood vessels traveling between the pelvis and the lower limb, including the sciatic nerve, the largest nerve in the human body. The pubis forms the anterior and medial portion of the hip bone and consists of a superior ramus, an inferior ramus, and a body that contains the pubic symphysis. The superior pubic ramus contributes to the acetabulum and provides attachment sites for muscles involved in hip adduction and flexion. The pubic tubercle, located on the superior ramus, serves as an attachment point for the inguinal ligament and marks an important landmark for hernia examinations. The inferior pubic ramus joins with the ramus of the ischium to complete the obturator foramen, the largest foramen in the human body. Despite its impressive size, this opening is almost completely closed by the obturator membrane in living individuals, with only a small opening remaining for the passage of the obturator nerve and vessels. The obturator foramen serves to reduce the weight of the hip bone while maintaining structural strength, demonstrating the principle of optimal design that characterizes many skeletal adaptations. The acetabulum represents the culmination of the three hip bone components, forming a deep, cup-shaped socket that articulates with the spherical head of the femur. The acetabular fossa, the deepest portion of the acetabulum, is filled with fat and ligamentous tissue rather than articular cartilage, while the horseshoe-shaped lunate surface provides the actual articulating surface for the femoral head. The acetabular notch interrupts the rim of the acetabulum inferiorly and is bridged by the transverse acetabular ligament, completing the socket while allowing for the passage of blood vessels that supply the femoral head. The pelvis as a complete structure demonstrates remarkable sexual dimorphism, with distinct differences between male and female pelves that reflect the different functional demands placed on these structures. The female pelvis shows numerous adaptations for childbirth, including a wider pelvic inlet, a more circular pelvic outlet, a wider subpubic angle, and more flared iliac wings. These modifications create a larger and more favorable birth canal while maintaining adequate strength for weight bearing and locomotion. The pelvic inlet, bounded by the sacral promontory posteriorly, the arcuate lines of the ilia laterally, and the superior margin of the pubic symphysis anteriorly, marks the boundary between the greater pelvis above and the lesser pelvis below. The greater pelvis supports the abdominal contents and provides attachment sites for abdominal muscles, while the lesser pelvis contains and protects the pelvic organs, including the bladder, reproductive organs, and rectum. The sacroiliac joints represent crucial articulations that demonstrate the complex balance between stability and mobility required for effective human locomotion. These joints are classified as synovial joints, but show characteristics of both synovial and fibrous joints, with extensive ligamentous support that severely limits movement while allowing for slight adjustments that help absorb shock and accommodate the changing demands of weight bearing and movement. The pubic symphysis, a cartilaginous joint connecting the two pubic bodies, provides additional stability to the pelvic ring while allowing for slight movements that help accommodate forces generated during locomotion. During pregnancy, hormonal changes increase the flexibility of this joint and other pelvic articulations, allowing for the slight expansion of the birth canal that facilitates childbirth. The overall architecture of the pelvic girdle creates a stable yet adaptable foundation that enables humans to maintain upright posture while walking, running, and performing the complex movements required for daily activities. The ring-like structure of the pelvis distributes forces effectively, while the deep hip sockets provide stable platforms for the powerful muscles that generate locomotor forces. This combination of stability and strength makes the pelvic girdle essential for the unique form of bipedal locomotion that characterizes human movement and distinguishes our species from our closest evolutionary relatives. 11. The appendicular skeleton, lower limb bones. The bones of the lower limb represent the ultimate expression of skeletal adaptation for bipedal locomotion, demonstrating remarkable structural modifications that enable humans to walk upright efficiently while maintaining the ability to run, jump, and perform complex movements that require both power and precision. From the massive femur, the longest and strongest bone in the human body, to the intricate arrangement of foot bones that provide both stability and propulsion during gait. Each element of the lower limb skeleton shows specific adaptations that reflect millions of years of evolutionary refinement for upright walking and running. The femur stands as a masterpiece of biological engineering, combining tremendous strength with optimal weight distribution to create a bone capable of withstanding forces several times body weight during vigorous activity. The proximal end of the femur features a spherical head that fits into the acetabulum of the hip bone, creating a ball and socket joint that provides excellent mobility while maintaining sufficient stability for weight bearing. The femoral head is covered with hyaline cartilage, except for a small depression called the fovea capitis, which serves as the attachment point for the ligamentum teres, a small ligament that carries blood vessels to the femoral head. The neck of the femur represents a critical structural element that positions the femoral head optimally within the acetabulum while providing adequate space for the extensive muscle attachments around the hip joint. The angle of inclination, approximately 125 degrees between the neck and shaft in adults, represents a compromise between mechanical efficiency and structural strength. This angle allows for adequate clearance during hip movement while maintaining reasonable lever arms for muscle action, though it also creates stress concentrations that make the femoral neck particularly susceptible to fracture, especially in elderly individuals with osteoporosis. The greater and lesser trochanters of the femur provide attachment sites for the powerful muscles that control hip movement and maintain pelvic stability during locomotion. The greater trochanter, a large, quadrangular projection on the lateral aspect of the proximal femur, serves as the insertion point for the gluteus medius, gluteus minimus, and several smaller muscles, crucial for maintaining hip stability during single limb support phases of walking. The lesser trochanter, smaller and located on the posteromedial aspect of the femur, provides attachment for the iliopsoas muscle, the primary hip flexor that is essential for lifting the thigh during walking and running. The shaft of the femur demonstrates a characteristic anterior bow that helps absorb shock and distribute forces during weight-bearing activities. The linea aspera, a prominent ridge running along the posterior aspect of the femoral shaft, provides attachment sites for the large muscles of the thigh, including the adductors and the hamstrings. This ridge becomes more pronounced in individuals who engage in regular physical activity, demonstrating the bone's ability to adapt its structure in response to mechanical demands. The distal end of the femur expands dramatically to form the knee joint with the tibia and patella, creating the largest and most complex joint in the human body. The medial and lateral condyles of the femur provide the articulating surfaces for the corresponding condyles of the tibia, while the patellar surface accommodates the patella during knee movements. The intercondylar notch separates the two condyles and provides space for the cruciate ligaments that are essential for knee stability. The tibia, the larger and more medial bone of the leg, serves as the primary weight-bearing element between the knee and ankle joints. The proximal end of the tibia features medial and lateral condyles that articulate with the corresponding condyles of the femur, creating the tibiofemoral articulation that forms the main component of the knee joint. The tibial plateau, formed by the superior surfaces of these condyles, provides a stable platform for weight transmission, while accommodating the menisci that help distribute loads and provide shock absorption during weight-bearing activities. The tibial tuberosity, a prominent projection on the anterior aspect of the proximal tibia, serves as the insertion point for the patellar ligament, which transmits the force generated by the quadriceps muscle group to extend the knee. This bony prominence is particularly well developed in individuals who engage in activities requiring frequent knee extension against resistance, such as jumping or kicking sports. The shaft of the tibia demonstrates a characteristic triangular cross-section with 3 distinct borders and 3 surfaces, providing optimal strength for weight-bearing while minimizing bone mass. The anterior border of the tibia, commonly known as the shin, lies just beneath the skin and can be easily palpated along its entire length. The medial surface is also subcutaneous and provides attachment sites for muscles involved in ankle and toe movements. The interosseous border of the tibia provides attachment for the interosseous membrane that connects the tibia and fibula along their length, helping to distribute forces between the two bones and providing additional surface area for muscle attachments. The posterior surface of the tibia contains the soleal line, which provides attachment for the soleus muscle, an important component of the calf muscle group responsible for plantar flexion and maintaining upright posture. The distal end of the tibia expands to form the primary weight-bearing component of the ankle joint, articulating with the talus bone of the foot. The medial malleolus, a prominent projection on the medial aspect of the distal tibia, extends downward to help form the socket that accommodates the talus, while also providing attachment points for ligaments that stabilize the ankle joint. The fibula, the smaller and more lateral bone of the leg, serves primarily as an attachment site for muscles rather than a major weight-bearing element, though it does contribute to ankle stability and bears approximately 10-15% of the load transmitted through the leg. The proximal end of the fibula, called the head, articulates with the lateral condyle of the tibia through a small synovial joint that allows for slight movements during ankle motion and weight-bearing activities. The neck of the fibula, located just below the head, represents an important anatomical landmark due to its relationship with the common fibular nerve, which wraps around this region as it travels from the thigh to innervate muscles in the anterior and lateral compartments of the leg. Injury to this area can result in foot drop, a condition characterized by weakness of dorsiflexion muscles. The shaft of the fibula provides attachment sites for numerous muscles involved in ankle and toe movements, including the peroneal muscles that are crucial for maintaining balance and providing lateral ankle stability. The fibula also contributes to the formation of the interosseous membrane and helps maintain the structural integrity of the leg during weight-bearing and locomotion. The lateral malleolus, formed by the distal end of the fibula, extends further distally than the medial malleolus and provides the lateral boundary of the ankle joint socket. This arrangement helps prevent excessive inversion of the foot while allowing for the normal range of ankle motion required for walking and running. The patella, or kneecap, represents the largest sesamoid bone in the human body and plays a crucial role in knee function by improving the mechanical advantage of the quadriceps muscle group. This triangular bone develops within the tendon of the quadriceps femoris muscle and articulates with the patellar surface of the femur, creating the patellofemoral joint that allows the patella to glide smoothly during knee flexion and extension. The posterior surface of the patella is covered with the thickest layer of hyaline cartilage found anywhere in the human body, reflecting the tremendous forces that pass through this joint during activities such as stair climbing, squatting, and jumping. The patella also helps distribute these forces over a larger surface area, reducing the stress concentration on the underlying femoral cartilage. The bones of the foot represent one of the most complex and functionally sophisticated arrangements of skeletal elements in the human body, working together to provide a stable base of support while maintaining the flexibility necessary for shock absorption and propulsion during locomotion. The 26 bones of each foot are arranged in 3 distinct groups: the tarsal bones of the hindfoot and midfoot, the metatarsal bones of the forefoot, and the phalanges of the toes. The 7 tarsal bones create the foundation of the foot architecture and are responsible for forming the characteristic arches that distinguish the human foot from that of other primates. The talus, the most superior tarsal bone, serves as the keystone of the foot by articulating with the tibia and fibula above to form the ankle joint, while also connecting with the calcaneus below and the navicular bone anteriorly to transmit forces throughout the foot. The calcaneus, the largest and most posterior tarsal bone, forms the heel and serves as the primary weight-bearing element during the stance phase of walking. This bone provides attachment for the Achilles tendon, the strongest tendon in the human body, which transmits the force generated by the calf muscles to produce plantar flexion and propulsion during walking and running. The calcaneus also features several important projections and processes that provide attachment sites for ligaments and muscles involved in foot stability and movement. The navicular bone, located on the medial aspect of the foot between the talus and the 3 cuneiform bones, plays a crucial role in maintaining the medial longitudinal arch of the foot. The navicular tuberosity provides attachment for the posterior tibial tendon, which helps support this arch and prevent excessive flattening during weight-bearing activities. The 3 cuneiform bones, tane, medial, intermediate, and lateral, articulate with the navicular posteriorly, and the first 3 metatarsals, anteriorly, forming part of the transverse arch of the foot and providing a stable foundation for the medial portion of the forefoot. The cuboid bone, located on the lateral aspect of the foot, articulates with the calcaneus posteriorly and the 4th and 5th metatarsals anteriorly, completing the midfoot structure. The 5 metatarsal bones form the structural framework of the forefoot and are numbered from medial to lateral, with the first metatarsal being the shortest and most robust, reflecting its crucial role in weight-bearing during the push-off phase of walking. Each metatarsal consists of a base that articulates with tarsal bones, a shaft that provides structural support, and a head that articulates with the corresponding proximal phalanx. The first metatarsal is particularly important for normal foot function, as it bears approximately twice the load of the other metatarsals during walking, and provides the primary propulsive force during push-off. The fifth metatarsal features a prominent tuberosity at its base that provides attachment for the peroneus brevis muscle and is frequently involved in fractures due to its exposed position. The phalanges of the toes follow the same general pattern as those in the hand, with 3 bones in each toe, except the great toe, which has only 2. However, the toe phalanges are much smaller and less mobile than their counterparts in the hand, reflecting their specialized role in providing stability and propulsion during locomotion rather than manipulation and grasping. The arrangement of foot bones creates 3 distinct arches that are essential for normal foot function: the medial longitudinal arch, the lateral longitudinal arch, and the transverse arch. The medial longitudinal arch is the highest and most functionally important, extending from the calcaneus to the heads of the first 3 metatarsals, and serving as a shock absorber during heel strike and a rigid lever during push-off. The lateral longitudinal arch is lower and more rigid than the medial arch, extending from the calcaneus to the heads of the 4th and 5th metatarsals, and providing stability during weight-bearing. The transverse arch runs across the midfoot and forefoot regions, helping to distribute forces across the width of the foot and contributing to the overall spring-like properties of foot architecture. These arch structures are maintained by a combination of bony shapes, ligamentous supports, and muscular actions, creating a dynamic system that can adapt to varying terrain and loading conditions while maintaining optimal function for bipedal locomotion. The evolution of these foot arches represents one of the most significant adaptations that enabled human ancestors to become efficient bipedal walkers and runners. 12. Joint classification and synovial joints. The classification and structure of joints represent fundamental concepts in understanding human movement and the remarkable engineering solutions that evolution has developed to address the competing demands of mobility and stability in the musculoskeletal system. Joints, or articulations, can be broadly classified based on their structural characteristics and the degree of movement they permit, with each type demonstrating specific adaptations that reflect their functional requirements within the overall framework of human locomotion and manipulation. Structurally, joints are classified into 3 major categories: fibrous joints, cartilaginous joints, and synovial joints. This classification system is based on the type of connective tissue that unites the bones at each articulation, with each structural type generally correlating with specific functional characteristics and movement capabilities. Understanding these structural relationships provides crucial insights into how different joints contribute to overall body function, and why certain joints are more susceptible to specific types of injuries or degenerative changes. Fibrous joints, also known as synarthroses, are characterized by the direct connection of bones through dense fibrous connective tissue, with no joint cavity present between the articulating elements. These joints are typically immovable or allow only slight movement, prioritizing stability over mobility to fulfill their specific functional roles. The 3 types of fibrous joints, sutures, syndesmoses, and gomphoses, each demonstrate unique structural features that reflect their particular mechanical requirements. Sutures represent the classic example of fibrous joints and are found exclusively in the skull, where they connect the various cranial and facial bones. These joints consist of thin layers of dense, fibrous connective tissue that allow for brain growth during infancy and childhood, while gradually ossifying during adulthood to create a solid, protective vault. The intricate interlocking edges of sutural joints provide exceptional strength while distributing forces across the entire skull structure. Syndesmoses involve bones connected by longer fibrous structures such as ligaments or interosseous membranes, allowing for slight movement while maintaining stability. The distal tibiofibular joint represents an excellent example of a syndesmosis, where the tibia and fibula are connected by strong ligaments that maintain the integrity of the ankle joint socket, while allowing for slight separation during dorsiflexion movements. Gomphoses are highly specialized, fibrous joints found only between teeth and their sockets in the maxilla and mandible. The periodontal ligament that connects each tooth to its socket allows for slight movement that helps absorb the tremendous forces generated during chewing, while maintaining secure tooth anchorage. Cartilaginous joints, or amphiarthroses, are characterized by the direct connection of bones through either hyaline cartilage or fibrocartilage, again with no joint cavity present. These joints typically allow more movement than fibrous joints but less than synovial joints, representing a compromise between stability and mobility that suits their specific functional roles. Synchondroses involve bones connected by hyaline cartilage and are typically temporary joints that eventually ossify as skeletal maturity is reached. The epiphyseal plates of long bones represent the most functionally significant synchondroses, allowing for longitudinal bone growth while maintaining structural integrity. Most synchondroses become synostoses, or bony joints, once growth is complete. Symphyses involve bones connected by fibrocartilage and typically persist throughout life, allowing for slight movement while providing substantial stability. The pubic symphysis and intervertebral discs represent the most clinically significant symphyses, demonstrating how these joints can accommodate forces while allowing for limited movement that contributes to overall spinal and pelvic function. Synovial joints, or diarthroses, represent the most complex and functionally diverse category of joints, characterized by the presence of a joint cavity filled with synovial fluid and surrounded by a joint capsule. These joints are designed primarily for movement, though they also demonstrate varying degrees of stability, depending on their specific structural features and functional requirements. The basic structure of synovial joints includes several key components that work together to facilitate smooth, pain-free movement while maintaining appropriate stability. The joint capsule represents the outer boundary of the synovial joint and consists of two distinct layers, an outer fibrous layer and an inner synovial membrane. The fibrous layer, composed of dense, irregular connective tissue, provides structural strength and helps maintain joint integrity during movement and loading. This layer is often reinforced by ligaments that provide additional stability and help guide joint movement in appropriate directions. The synovial membrane lines the inner surface of the joint capsule and all non-articulating surfaces within the joint cavity, including ligaments and tendons that pass through the joint space. This specialized membrane produces synovial fluid, a viscous lubricant that reduces friction between articulating surfaces and provides nutrition to the avascular articular cartilage through diffusion. Articular cartilage covers the ends of bones within synovial joints and provides a smooth, low-friction surface that can withstand tremendous compressive forces while allowing for smooth movement. This specialized hyaline cartilage is avascular and receives its nutrition through diffusion from synovial fluid, making it particularly dependent on regular movement for maintaining health and preventing degeneration. Many synovial joints contain additional structures that enhance their function or provide specialized capabilities. Menisci, found in joints such as the knee and temporomandibular joint, are crescent-shaped fibrocartilage structures that help distribute forces, deepen joint sockets, and provide additional stability during movement. Articular discs serve similar functions in joints, such as the sternoclavicular and acromioclavicular joints. Bursae represent small, synovial fluid-filled sacs that are located between structures that move relative to each other, such as tendons and bones, or muscles and bones. These structures help reduce friction and prevent wear during repetitive movements, though they can become inflamed and painful in conditions known as bursitis. The classification of synovial joints is based primarily on the shapes of the articulating surfaces and the types of movements they permit. Ball and socket joints, such as the shoulder and hip joints, feature a spherical head fitting into a cup-shaped socket, allowing for movement in all three planes of motion, including flexion-extension, abduction-adduction, and rotation. Hinge joints, such as the elbow and ankle joints, feature articulating surfaces that are shaped like a cylinder, fitting into a corresponding trough, allowing primarily for flexion and extension movements in a single plane. These joints often feature strong collateral ligaments that prevent unwanted side-to-side movement while allowing for the primary hinge motion. Pivot joints, such as the atlantoaxial joint and the proximal radioulnar joint, feature a rounded process fitting into a ring formed by bone and ligament, allowing for rotational movement around a single axis. These joints are specialized for rotation while maintaining stability in other directions. Condylar joints, such as the metacarpophalangeal joints, feature oval-shaped condyles fitting into corresponding depressions, allowing for movement in two planes, but preventing rotation. These joints provide an excellent compromise between mobility and stability for digits and similar structures. Saddle joints, found primarily at the base of the thumb, feature articulating surfaces that are saddle-shaped, with each surface being concave in one direction and convex in the perpendicular direction. This arrangement allows for movement in two planes while providing inherent stability through the interlocking shapes. Gliding or plane joints, such as those found between carpal bones and tarsal bones, feature relatively flat articulating surfaces that allow for small sliding movements in multiple directions. These joints are crucial for the complex movements of the hands and feet, while maintaining overall structural integrity. The synovial fluid that fills synovial joint cavities represents a remarkable biological lubricant that demonstrates properties superior to any artificial lubricant yet developed. This fluid is produced by the synovial membrane and consists of a filtrate of blood plasma combined with hyaluronic acid and other glycosaminoglycans that give it its characteristic viscous properties. The lubricating properties of synovial fluid change with the rate of movement, becoming less viscous during rapid movements to allow for easy motion, while becoming more viscous during slow movements to provide better protection against wear. This thixotropic behavior, combined with the extremely smooth surfaces of healthy articular cartilage, creates a system with a coefficient of friction lower than that achieved by ice sliding on ice. Understanding the classification and structure of joints provides the foundation for comprehending how the musculoskeletal system achieves its remarkable combination of stability and mobility, enabling the complex movements required for human locomotion, manipulation, and the countless activities that characterize human behavior and capabilities. 13. Types of synovial joints and movement. The remarkable diversity of synovial joint types reflects the evolutionary solutions to the complex mechanical challenges faced by the human musculoskeletal system in balancing mobility with stability across different anatomical regions and functional demands. Each type of synovial joint demonstrates specific structural adaptations that enable particular movement patterns while providing appropriate stability for their role in human locomotion and manipulation. Understanding these joint types and their characteristic movements provides crucial insights into how the body generates and controls the complex patterns of motion required for daily activities. Ball and socket joints represent the pinnacle of synovial joint mobility, featuring a spherical or semi-spherical head that fits into a correspondingly shaped socket or acetabulum. This design permits movement in all three anatomical planes, making these joints triaxial and providing the greatest range of motion of any joint type. The shoulder joint, formed by the articulation between the humeral head and the glenoid cavity of the scapula, exemplifies the mobility-focused design of ball and socket joints, sacrificing stability to achieve an extraordinary range of motion that enables the arm to reach virtually any point in space around the body. The hip joint demonstrates a different approach to ball and socket design, featuring a much deeper socket and tighter capsular constraints that prioritize stability while maintaining substantial mobility. The deeper acetabulum and strong ligamentous support of the hip joint reflect its primary role in weight-bearing and locomotion, where stability is crucial for effective force transmission and injury prevention. Despite these stability features, the hip joint still permits flexion-extension, abduction-adduction, and rotation movements that are essential for walking, running, and complex lower limb activities. The trade-off between mobility and stability in ball and socket joints illustrates a fundamental principle of joint design, where increased range of motion typically comes at the expense of inherent stability, requiring greater reliance on muscular control and ligamentous support to maintain joint integrity. This relationship explains why the shoulder joint is the most frequently dislocated major joint in the body, while hip dislocations are relatively rare despite the similar basic joint structure. Hinge joints represent the opposite end of the mobility-stability spectrum, featuring articulating surfaces designed like mechanical hinges that permit movement primarily in a single plane. The elbow joint, formed by the articulation between the trochlea of the humerus and the trochlear notch of the ulna, demonstrates the classic hinge joint design with its pulley-like surfaces that guide flexion and extension movements while preventing unwanted rotation or side-to-side motion. The structural features that characterize hinge joints include closely conforming, articulating surfaces, strong collateral ligaments that prevent abduction and adduction, and often additional bony constraints that limit movement to the desired plane. The ankle joint provides another excellent example of hinge joint design, though it demonstrates slight modifications that allow for minimal amounts of rotation and side-to-side movement that are necessary for walking on uneven surfaces. The apparent simplicity of hinge joint movement belies the complex coordination required to produce smooth, controlled motion throughout the range of flexion and extension. The changing geometry of hinge joint surfaces means that the axis of rotation is not fixed, but shifts slightly throughout the range of motion, requiring sophisticated neuromuscular control to maintain smooth movement patterns and appropriate force distribution across the joint surfaces. Pivot joints represent specialized synovial joints designed specifically for rotational movement around a single axis, featuring a rounded process that rotates within a ring formed by bone and ligament. The atlantoaxial joint, formed by the dens of the axis rotating within the ring formed by the atlas and the transverse ligament, provides approximately half of the total rotation available in the cervical spine, and demonstrates the highly specialized nature of pivot joint design. The proximal radioulnar joint represents another crucial pivot joint that enables pronation and supination of the forearm, movements that are essential for optimal hand positioning during manipulation tasks. The head of the radius rotates within the radial notch of the ulna and the annular ligament, allowing the radius to cross over the ulna during pronation while maintaining stable elbow joint function. Condylar joints, also known as ellipsoidal joints, feature oval-shaped condyles that articulate with corresponding elliptical cavities, creating joints that allow movement in two planes while preventing rotation. The metacarpophalangeal joints of the fingers exemplify condylar joint design, permitting flexion-extension and abduction-adduction while preventing rotation that could compromise grip strength and hand stability. The radiocarpal joint at the wrist represents a modified condylar joint that demonstrates how basic joint types can be adapted for specific functional requirements. The elliptical distal end of the radius articulates with the scaphoid and lunate bones of the wrist, allowing for flexion extension and radial ulnar deviation, while the complex arrangement of carpal bones provides additional movement capabilities through their numerous intercarpal articulations. Saddle joints represent a unique synovial joint type, found primarily at the base of the thumb, where the trapezium and first metacarpal create articulating surfaces that are concave in one direction and convex in the perpendicular direction. This saddle-shaped configuration allows for movement in two planes while providing inherent stability through the interlocking surface geometry, making it ideally suited for the specialized movements required for thumb opposition. The carpometacarpal joint of the thumb enables the remarkable dexterity that characterizes human hand function, allowing the thumb to touch each of the other digits while maintaining strength for powerful grip activities. This joint permits flexion-extension, abduction-adduction, and a limited amount of rotation that combines to produce the complex circumduction movements necessary for opposition. Gliding or plane joints feature relatively flat or slightly curved, articulating surfaces that allow for small sliding movements in multiple directions without significant rotation or angular motion. These joints are found primarily in the hands and feet, where the numerous carpal and tarsal bones articulate through plane joints that collectively provide substantial mobility while maintaining structural integrity. The intercarpal and intertarsal joints demonstrate how multiple plane joints can work together to produce complex movement patterns that would be impossible with single, more mobile joints. The small movements at each individual plane joint sum together to produce the significant overall mobility of the hand and foot, while distributing forces across multiple articulations to prevent excessive stress on any single joint. The temporomandibular joint represents a unique example of joint design that combines features of different joint types to accommodate the complex functional demands of mastication, speech, and facial expression. This joint functions as both a hinge joint for opening and closing movements and a gliding joint for the protrusive and lateral movements necessary for effective chewing. The movement capabilities of synovial joints are further enhanced by the coordinated action of multiple joints working together in kinetic chains that amplify the movement potential of individual articulations. The shoulder complex, consisting of the glenohumeral, acromioclavicular, sternoclavicular, and scapulothoracic articulations, demonstrates how multiple joints can work together to produce movement patterns that exceed the capabilities of any individual joint. The concept of degrees of freedom provides a useful framework for understanding joint movement capabilities, with each degree of freedom representing an independent direction of movement. Monaxial joints, such as hinge and pivot joints, permit movement around a single axis, while biaxial joints, such as condylar and saddle joints, allow movement around two axes, and triaxial joints, such as ball and socket joints, permit movement around three axes. The movement patterns produced by different joint types are also influenced by the surrounding musculature, which must provide both the forces necessary for movement and the stabilizing forces required to control unwanted motion. The relationship between joint structure and muscle architecture demonstrates the intimate connection between the skeletal and muscular systems in producing controlled, purposeful movement. Understanding the types of synovial joints and their movement characteristics provides the foundation for comprehending how the human body achieves its remarkable movement capabilities while maintaining the stability necessary for effective function in the complex mechanical environment that characterizes human activities and behavior. 14. Movement terminology and biomechanics. The systematic description and analysis of human movement requires a precise vocabulary that can accurately describe the complex three-dimensional motions that characterize human locomotion and manipulation. This standardized terminology, developed over centuries of anatomical and biomechanical study, provides the foundation for understanding how the musculoskeletal system generates and controls movement while maintaining appropriate stability and coordination. The biomechanical principles that govern human movement reflect the fundamental laws of physics applied to biological systems, creating a framework for understanding how forces, leverage, and momentum interact to produce the remarkable movement capabilities that distinguish human motor function. The anatomical position serves as the fundamental reference point for all movement descriptions, establishing a standardized orientation that allows for consistent communication about body position and movement direction. In the anatomical position, the individual stands upright with feet parallel and shoulder width apart, arms at the sides with palms facing forward, and head facing straight ahead. This reference position defines the neutral starting point from which all movements are described, ensuring that movement terminology remains consistent regardless of the observer's perspective or the individual's starting position. The anatomical planes provide the three-dimensional framework within which all human movements occur, with each plane representing a flat surface that divides the body into specific sections. The sagittal plane divides the body into left and right halves, and contains movements such as flexion and extension that occur around a mediolateral axis. The frontal plane, also called the coronal plane, divides the body into anterior and posterior portions, and contains movements such as abduction and adduction that occur around an anteroposterior axis. The transverse plane divides the body into superior and inferior portions and contains rotational movements that occur around a longitudinal axis. Flexion represents a movement that decreases the angle between two body segments, typically involving the approximation of anterior surfaces in the sagittal plane. This fundamental movement pattern is exemplified by bending the elbow to bring the hand toward the shoulder, bending the knee to bring the heel toward the buttock, or bending the trunk forward at the waist. Flexion movements are generally associated with the action of flexor muscles and represent one of the most common and functionally important movement patterns in human locomotion and manipulation. Extension represents the opposite of flexion, involving an increase in the angle between two body segments and typically moving body parts away from each other in the sagittal plane. Extension movements include straightening the elbow, knee, or spine, and are generally produced by extensor muscles that work in opposition to flexor muscles. The coordination between flexion and extension movements creates the reciprocal muscle actions that characterize most human movement patterns and provide the basis for efficient locomotion and manipulation. Hyperextension occurs when extension movement continues beyond the normal anatomical position, creating angles greater than 180 degrees between body segments. This movement is possible at certain joints such as the shoulder, hip, and spine, but is limited or prevented at others such as the elbow and knee by bony constraints and ligamentous restrictions. Understanding the normal limits of hyperextension is crucial for preventing injury and optimizing athletic performance. Abduction represents movement away from the midline of the body in the frontal plane, such as raising the arm to the side or spreading the fingers apart. This movement pattern is produced by abductor muscles and is essential for creating the wide base of support and limb positioning necessary for many functional activities. The reference point for abduction varies depending on the body part, with limb abduction referenced to the body's midline, while finger and toe abduction is referenced to the midline of the hand or foot respectively. Adduction represents the opposite of abduction, involving movement toward the midline of the body in the frontal plane. This movement pattern brings body parts closer to the central axis and is produced by adductor muscles that work in coordination with abductors to control limb positioning and provide stability during various activities. The balance between abduction and adduction is crucial for maintaining appropriate joint alignment and preventing excessive stress on joint structures. Rotation represents movement around the longitudinal axis of a body segment and occurs in the transverse plane. Internal rotation, also called medial rotation, involves turning toward the midline of the body, while external rotation, also called lateral rotation, involves turning away from the midline. Rotational movements are essential for optimal positioning of the limbs during functional activities and contribute significantly to the overall range of motion available at ball and socket joints. Circumduction represents a complex movement pattern that combines flexion, extension, abduction, and adduction in sequence to create a circular or cone-shaped path of motion. This movement is possible at ball and socket joints and some condylar joints, allowing for the tracing of circles or ellipses in space without rotation around the longitudinal axis. Circumduction demonstrates the remarkable versatility of certain joint types and their ability to produce complex movement patterns through the coordination of multiple muscle groups. Pronation and supination represent specialized rotational movements that occur primarily in the forearm, allowing for optimal hand positioning during manipulation tasks. Pronation involves rotation of the forearm so that the palm faces posteriorly or downward, while supination involves rotation so that the palm faces anteriorly or upward. These movements are produced by the coordinated action of muscles acting on the proximal and distal radial nerve joints and are essential for activities such as turning doorknobs, using screwdrivers, or pouring liquids. Inversion and eversion represent specialized movements of the foot that occur primarily at the subtalar joint, allowing for adaptation to uneven terrain during locomotion. Inversion involves turning the sole of the foot medially toward the midline, while eversion involves turning the sole laterally away from the midline. These movements are crucial for maintaining balance and preventing ankle injuries during activities on irregular surfaces. Dorsiflexion and plantarflexion represent the primary movements at the ankle joint, with dorsiflexion involving movement of the foot toward the anterior leg surface, and plantarflexion involving movement away from the leg in the direction of the plantar surface. These movements are essential for walking, running, and maintaining balance, with dorsiflexion occurring during the swing phase of gait and early stance phase, while plantarflexion provides propulsion during push-off. Protraction and retraction represent movements in the transverse plane that involve moving body parts forward or backward relative to the coronal plane. These terms are commonly applied to movements of the shoulder girdle, where protraction involves moving the scapulae away from the spine, while retraction involves moving them toward the spine. These movements are essential for optimal arm positioning and contribute to the overall mobility of the shoulder complex. Elevation and depression represent movements in the frontal plane that involve raising or lowering body parts relative to their normal position. These movements are commonly described for the shoulder girdle, where elevation involves raising the shoulders toward the ears, while depression involves lowering them. The mandible also demonstrates elevation and depression movements during chewing and speaking. The biomechanical analysis of human movement requires understanding the principles of leverage and mechanical advantage that govern how forces are transmitted through the musculoskeletal system. The human body contains numerous examples of lever systems, where bones act as rigid levers, joints serve as fulcrums, and muscles provide the effort forces that overcome resistance forces, such as body weight or external loads. First-class levers have the fulcrum positioned between the effort and resistance forces, creating a system that can provide either mechanical advantage or speed advantage, depending on the relative distances from the fulcrum. The atlanto-occipital joint represents a classic first-class lever, where the joint serves as the fulcrum. The weight of the head provides the resistance, and the posterior neck muscles provide the effort force to maintain head position. Second-class levers have the resistance positioned between the fulcrum and effort, always providing mechanical advantage by allowing smaller effort forces to overcome larger resistance forces. The action of rising up on the toes represents a second-class lever system, where the metatarsophalangeal joints serve as the fulcrum. Body weight provides the resistance, and the calf muscles provide the effort force. Third-class levers have the effort position between the fulcrum and resistance, always favoring speed and range of motion over force advantage. The majority of muscular actions in the human body represent third-class lever systems, such as elbow flexion, where the elbow joint serves as the fulcrum. The biceps muscle provides the effort, and any weight in the hand or the weight of the forearm provides the resistance. Understanding movement terminology and biomechanical principles provides the foundation for analyzing human movement patterns, identifying movement dysfunctions, designing effective exercise programs, and preventing injuries through proper movement mechanics and load distribution throughout the musculoskeletal system. 15. Skeletal muscle structure and function. Skeletal muscle tissue represents one of the most remarkable and specialized tissues in the human body, demonstrating extraordinary adaptability and functional sophistication that enables the generation of precise and controlled forces across an enormous range of magnitudes and speeds. From the delicate movements required for fine motor control to the explosive power needed for athletic performance, skeletal muscle tissue demonstrates structural and functional characteristics that reflect millions of years of evolutionary refinement for movement generation and control. Understanding the hierarchical organization of skeletal muscle, from the molecular level to the whole muscle, provides crucial insights into how this tissue accomplishes its diverse functional roles. Skeletal muscle tissue is characterized by several distinctive features that distinguish it from cardiac and smooth muscle types and reflect its specialized role in voluntary movement generation. The most obvious characteristic is the presence of cross striations, alternating light and dark bands that give skeletal muscle its striated appearance when viewed under microscopic examination. These striations reflect the highly organized arrangement of contractile proteins that enable muscle contraction through the sliding filament mechanism. The multinucleated nature of skeletal muscle fibers represents another distinctive characteristic that reflects the developmental history and metabolic demands of this tissue. Individual muscle fibers, which can extend the entire length of a muscle, are formed during development through the fusion of multiple myoblasts, creating elongated cells that may contain hundreds or even thousands of nuclei distributed along their length. This multinucleated arrangement provides the genetic machinery necessary to support the high protein synthesis demands required for maintaining and repairing the extensive contractile apparatus. The voluntary nature of skeletal muscle contraction distinguishes this tissue from cardiac and smooth muscle types, and reflects the direct neural control that conscious motor commands exert over skeletal muscle function. This voluntary control is mediated through the somatic nervous system, with motor neurons extending directly from the central nervous system to innervate individual muscle fibers through specialized synapses called neuromuscular junctions. The connective tissue organization of skeletal muscle demonstrates a hierarchical arrangement that provides both structural support and functional coordination throughout the muscle structure. The endomysium represents the innermost layer of connective tissue that surrounds individual muscle fibers, providing a supportive matrix that contains capillaries, nerve endings. The connective tissue organization of skeletal muscle demonstrates a hierarchical arrangement that provides both structural support and functional coordination throughout the muscle structure. The endomysium represents the innermost layer of connective tissue that surrounds individual muscle fibers, providing a supportive matrix that contains capillaries, nerve endings, and the extracellular components necessary for fiber nutrition and waste removal. This delicate connective tissue layer allows for individual fiber contraction while maintaining the structural integrity necessary for coordinated muscle function. The paramecium represents the intermediate layer of connective tissue that groups muscle fibers into bundles called fascicles, typically containing between 10 and 100 individual fibers, depending on the muscle's functional requirements. The fascicular organization allows for the independent control of different regions within a muscle and contributes to the graded nature of muscle contraction by enabling the selective recruitment of different fascicles based on the force requirements of a particular task. The epimysium forms the outermost connective tissue layer that surrounds the entire muscle, providing structural continuity and force transmission between the muscle and its tendinous attachments. This robust connective tissue layer merges with the tendon or aponeurosis at the muscle's ends, creating a continuous pathway for force transmission from the contractile elements to the skeletal system. The arrangement of fascicles within muscles varies considerably depending on the functional requirements of each muscle, creating distinct architectural patterns that reflect the relationship between force generation and range of motion. Parallel fascicle arrangement, found in muscles such as the sartorius and biceps brachii, maximizes the range of motion and speed of contraction by aligning fascicles parallel to the muscle's line of action. This arrangement is optimal for muscles that need to produce large changes in length during contraction. Pennate fascicle arrangements, where fascicles insert into tendons at angles rather than parallel to the line of action, maximize force production by allowing more fascicles to be packed into a given muscle volume. Unipennate muscles, such as the extensor digitorum longus, have fascicles arranged on one side of a central tendon, while bipennate muscles, such as the rectus femoris, have fascicles arranged on both sides of a central tendon. Multipennate muscles, such as the deltoid, have fascicles arranged in multiple directions around several tendons, creating complex force vectors that can be selectively activated for different movement patterns. Convergent fascicle arrangements, found in muscles such as the pectoralis major and latissimus dorsi, have fascicles that spread out from a narrow attachment to a broader insertion, allowing for both force production and versatility in the direction of pull. Circular fascicle arrangements, found in sphincter muscles such as those around the mouth and eyes, create the ability to decrease the diameter of openings through concentric contraction, The individual muscle fiber represents the basic functional unit of skeletal muscle and demonstrates remarkable structural complexity that enables the conversion of chemical energy into mechanical work. Each muscle fiber is surrounded by a specialized cell membrane called the sarcolemma, which maintains the fiber's structural integrity while regulating the passage of ions and nutrients necessary for contraction and metabolism. The sarcoplasm, the cytoplasm of muscle fibers, contains the specialized organelles and proteins necessary for muscle function, including numerous mitochondria that provide the ATP required for contraction, an extensive sarcoplasmic reticulum that stores and releases calcium ions for contraction regulation, and thousands of myofibrils that contain the actual contractile apparatus. Myofibrils represent the contractile organelles within muscle fibers and consist of repeating functional units called sarcomeres, arranged in series along the length of the fiber. Each myofibril may contain thousands of sarcomeres, with the total number depending on the fiber length and the muscle's functional requirements. The parallel arrangement of multiple myofibrils within each fiber allows for the summation of forces generated by individual sarcomeres. The sarcomere represents the fundamental contractile unit of skeletal muscle and extends from one Z-disc to the next, along the length of the myofibril. The characteristic striation pattern of skeletal muscle results from the highly organized arrangement of thick and thin filaments within each sarcomere, with the A bands containing thick filaments and the I bands containing only thin filaments. The thick filaments consist primarily of the protein myosin, with each myosin molecule containing a tail region that forms the backbone of the filament, and globular heads that extend outward to interact with thin filaments during contraction. The myosin heads contain both actin binding sites and ATP activity, enabling them to bind to thin filaments and generate force through conformational changes powered by ATP hydrolysis. The thin filaments consist primarily of the protein actin, arranged in a double helical structure that provides binding sites for myosin heads during contraction. The regulatory proteins tropomyosin and troponin are associated with thin filaments and control the interaction between actin and myosin based on the presence or absence of calcium ions. The sliding filament mechanism of muscle contraction represents one of the most elegant examples of molecular machinery in biological systems, converting the chemical energy stored in ATP into the mechanical work necessary for movement. During contraction, the thick and thin filaments slide past each other without changing length, causing the sarcomere to shorten and generating tension that is transmitted through the connective tissue system to produce movement at joints. The excitation-contraction coupling process represents the link between electrical stimulation of the muscle fiber and the initiation of the sliding filament mechanism. When a motor neuron stimulates a muscle fiber at the neuromuscular junction, the resulting action potential spreads across the sarcolemma and penetrates into the fiber interior through the tubule system, a network of membrane invaginations that ensures rapid and uniform activation throughout the fiber. The tubule system communicates with the sarcoplasmic reticulum, an extensive network of membranous channels that stores calcium ions in the relaxed muscle. The arrival of the action potential at the tubules triggers the release of calcium ions from the sarcoplasmic reticulum through specialized calcium release channels, rapidly increasing the calcium concentration in the sarcoplasm surrounding the myofibrils. The released calcium ions bind to troponin molecules on the thin filaments, causing a conformational change that moves tropomyosin away from the myosin binding sites on actin. This exposes the binding sites and allows myosin heads to form cross bridges with actin, initiating the power stroke that generates force and shortening. The cross-bridge cycle represents the cyclic interaction between myosin and actin that generates force during muscle contraction. The cycle begins with the binding of ATP to the myosin head, causing it to detach from actin and assume a high-energy configuration through ATP hydrolysis. The energized myosin head then binds to a new actin binding site, forms a cross-bridge, and undergoes the power stroke that generates force and movement. The cycle concludes with the binding of a new ATP molecule, which allows the cross bridge to detach and prepare for another cycle. The termination of muscle contraction occurs when neural stimulation ceases and calcium ions are actively transported back into the sarcoplasmic reticulum by calcium pumps. The decrease in calcium concentration allows tropomyosin to resume its blocking position on the thin filaments, preventing further cross-bridge formation and allowing the muscle to relax and return to its resting length. The force-generating capacity of skeletal muscle depends on several factors, including the number of cross-bridges that can be formed simultaneously, the frequency of cross-bridge cycling, and the mechanical properties of the muscle architecture. The length-tension relationship demonstrates that muscles generate maximum force when sarcomeres are at their optimal length, where there is maximum overlap between thick and thin filaments for cross-bridge formation. The force-velocity relationship shows that muscles can generate maximum force during isometric contractions, when there is no change in muscle length, while force production decreases as the speed of contraction increases during concentric contractions. During eccentric contractions, where muscles are forced to lengthen while contracting, force production can exceed that achieved during isometric contractions, though this type of contraction is associated with greater risk of muscle damage. Understanding skeletal muscle structure and function provides the foundation for comprehending how the neuromuscular system generates and controls the forces necessary for human movement, while adapting to the varying demands of different activities and environmental conditions. 16. Muscle contraction mechanism. The molecular mechanism of muscle contraction represents one of the most thoroughly studied and elegantly understood processes in cell biology, demonstrating how specialized proteins convert chemical energy into mechanical work with remarkable efficiency and precision. The sliding filament theory, first proposed in the 1950s and subsequently refined through decades of research, provides a comprehensive framework for understanding how sarcomeres generate force and produce muscle shortening through the cyclic interaction of myosin and actin filaments. The fundamental insight of the sliding filament theory is that muscle contraction occurs not through the shortening of individual protein filaments, but through the relative sliding of thick and thin filaments past each other within the sarcomere. This mechanism allows muscles to generate force while maintaining the structural integrity of the contractile proteins and enables the precise control of force production through the regulation of cross-bridge formation and cycling. The thick filaments, composed primarily of myosin molecules, demonstrate a highly organized structure that positions myosin heads optimally for interaction with thin filaments. Each myosin molecule consists of 2 heavy chains and 4 light chains arranged to form a structure with a long tail region and 2 globular heads. The tail regions of approximately 300 myosin molecules align to form the backbone of each thick filament, while the heads project outward in a helical arrangement that ensures they can interact with multiple thin filaments. The myosin heads contain 2 crucial functional domains: an actin binding site that enables attachment to thin filaments, and an ATPase domain that hydrolyzes ATP to provide the energy necessary for force generation. The spatial relationship between these domains allows the myosin head to undergo conformational changes that translate the chemical energy released by ATP hydrolysis into mechanical work through the power stroke mechanism. The thin filaments demonstrate an equally sophisticated organization that enables precise regulation of the interaction between actin and myosin. The backbone of thin filaments consists of filamentous actin, formed by the polymerization of globular actin monomers into a double helical structure. Each actin monomer contains a myosin binding site that, under appropriate conditions, can form cross bridges with myosin heads to generate force. The regulatory proteins tropomyosin and troponin are intimately associated with thin filaments and provide the molecular switch that controls muscle contraction in response to calcium ions. Tropomyosin molecules lie in the groove between the 2 actin strands and, in the relaxed state, physically block the myosin binding sites on actin, preventing cross-bridge formation, even in the presence of ATP. The troponin complex consists of three subunits that work together to regulate tropomyosin position. Troponin C contains the calcium binding sites that detect changes in calcium concentration. Troponin I inhibits actin-myosin interaction in the absence of calcium, and troponin T binds to tropomyosin to transmit conformational changes throughout the regulatory system. The excitation-contraction coupling process begins with the arrival of an action potential at the muscle fiber membrane, initiating a cascade of events that rapidly increases the calcium concentration throughout the sarcoplasm. The action potential spreads across the sarcolemma and penetrates deep into the muscle fiber through the transverse tubule system, ensuring that all parts of the fiber receive the excitation signal simultaneously. The transverse tubules, formed by deep invaginations of the sarcolemma, create a network of membrane-lined channels that bring the electrical signal into close proximity with the sarcoplasmic reticulum throughout the fiber. At specialized junctions called triads, the tubules come into intimate contact with terminal cisternae of the sarcoplasmic reticulum, creating the structural arrangement necessary for rapid signal transmission from the surface membrane to the calcium storage system. The sarcoplasmic reticulum represents a specialized form of endoplasmic reticulum that has evolved specifically for calcium storage and release in muscle fibers. In the relaxed state, calcium pumps in the sarcoplasmic reticulum membrane actively transport calcium ions from the sarcoplasm into the storage compartments, maintaining a very low calcium concentration around the myofibrils and preventing unwanted muscle contraction. The arrival of the action potential at the tubule membrane triggers the opening of voltage-sensitive calcium release channels in the sarcoplasmic reticulum membrane, causing a massive and rapid release of stored calcium ions into the sarcoplasm. This calcium release increases the sarcoplasmic calcium concentration from approximately 0.1 micromolar to 10 micromolar within milliseconds, providing the signal necessary for contraction initiation. The released calcium ions bind to specific sites on troponin C, causing a conformational change in the troponin complex that is transmitted to tropomyosin through troponin T. This conformational change causes tropomyosin to shift position in the groove between actin strands, exposing the myosin binding sites on actin and enabling cross-bridge formation. The cross-bridge cycle represents the mechanical core of muscle contraction, consisting of a series of conformational changes in the myosin head that generate force and produce relative sliding between thick and thin filaments. The cycle begins with the myosin head in a high-energy state, with ADP and inorganic phosphate bound to the myosin ATPase site and the head positioned away from the actin filament. The binding of the energized myosin head to the exposed binding site on actin forms the initial cross bridge and triggers the release of inorganic phosphate from the myosin ATPase site. This phosphate release is associated with a conformational change in the myosin head that constitutes the power stroke, rotating the head and pulling the thin filament toward the center of the sarcomere. The power stroke is followed by the release of ADP from the myosin head, leaving the myosin tightly bound to actin in what is called the rigor state. The binding of a new ATP molecule to the myosin ATPase site causes the myosin head to detach from actin and return to its original position, while the hydrolysis of ATP to ADP and inorganic phosphate re-energizes the myosin head for another cycle. The cyclic repetition of cross-bridge formation, power stroke, and detachment continues as long as calcium ions remain bound to troponin and ATP is available for cross-bridge cycling. The summation of forces generated by thousands of cross-bridges, cycling asynchronously throughout each sarcomere, produces the macroscopic force that characterizes muscle contraction. The regulation of contraction termination occurs through the removal of calcium ions from the sarcoplasm, primarily through the action of calcium pumps in the sarcoplasmic reticulum membrane. These ATP-powered pumps actively transport calcium ions from the sarcoplasm back into the sarcoplasmic reticulum storage compartments, reducing the calcium concentration and allowing tropomyosin to resume its blocking position on the thin filaments. The energy requirements for muscle contraction are substantial. with ATP consumption occurring at multiple steps in the process, including cross-bridge cycling, calcium pumping, and maintenance of ionic gradients across cell membranes. The immediate source of ATP for contraction comes from the muscle fibers' existing ATP stores, but these are rapidly depleted and must be regenerated through various metabolic pathways. The phosphocreatine system provides the most immediate mechanism for ATP regeneration, using the high-energy phosphate bonds stored in phosphocreatine to rapidly rephosphorylate ADP to ATP through the action of creatine kinase. This system can maintain ATP levels for approximately 10-15 seconds of maximal muscle activity and is particularly important for activities requiring high power output over short durations. Glycolysis provides ATP through the breakdown of glucose or glycogen and can operate under both aerobic and anaerobic conditions, though anaerobic glycolysis results in the accumulation of lactate that may contribute to muscle fatigue. Oxidative metabolism provides the most efficient mechanism for ATP production through the complete oxidation of carbohydrates and fats in the mitochondria, but this process requires adequate oxygen delivery and is limited by the capacity of the cardiovascular system. The remarkable efficiency of the muscle contraction mechanism, combined with its precise regulation and adaptability, enables skeletal muscle to meet the diverse energy and force requirements of human movement, while maintaining the molecular machinery necessary for sustained activity throughout life. 17. Major muscle groups. Axial muscles. The axial muscles represent the foundational muscular system of the human body, encompassing the muscles that move and stabilize the head, neck, and trunk while providing essential support for upright posture and enabling the complex movements required for respiration, communication, and spinal mobility. These muscles demonstrate remarkable specialization for their diverse functional roles, from the delicate muscles of facial expression that enable non-verbal communication to the powerful muscles of the back and abdomen that maintain spinal stability and generate the forces necessary for lifting and carrying activities. The muscles of the head and neck demonstrate extraordinary diversity and specialization, reflecting the complex functional requirements of structures involved in vision, hearing, chewing, swallowing, breathing, and communication. The muscles of facial expression, innervated by the facial nerve, represent a unique group of muscles that insert into skin rather than bone, enabling the subtle and complex movements that characterize human facial expression and nonverbal communication. The frontalis muscle, located in the forehead region, elevates the eyebrows and creates horizontal wrinkles across the forehead during expressions of surprise or concern. This muscle works in conjunction with the occipitalis muscle at the back of the head through the epicranial aponeurosis, creating a muscular system that can move the scalp and contribute to facial expression. The orbicularis oculi muscle surrounds the eye socket and consists of orbital and palpebral portions that enable both gentle and forceful eye closure. The orbital portion produces the tight closure associated with squinting or protecting the eyes from bright light, while the palpebral portion enables the gentle closure associated with blinking and maintaining eye moisture. The muscles around the mouth demonstrate particular complexity and importance for human communication and feeding behaviors. The orbicularis oris muscle forms the muscular foundation of the lips and enables the lip movements necessary for speech, eating, and facial expression. This muscle works in coordination with numerous other muscles that insert into the lips from various directions, creating the precise control necessary for articulation and the complex lip movements required for different phonetic sounds. The buccinator muscle, located in the cheek region, plays crucial roles in both mastication and speech production. During chewing, this muscle compresses the cheek against the teeth to prevent food from accumulating in the oral vestibule and assists in moving food onto the chewing surfaces of the teeth. During speech and wind instrument playing, the buccinator helps control air pressure within the oral cavity and contributes to the precise lip and cheek movements necessary for sound production. The muscles of mastication represent some of the strongest muscles in the human body relative to their size, generating the tremendous forces necessary for crushing and grinding food during chewing. The temporalis muscle, arising from the temporal fossa on the side of the skull, provides powerful closing forces for the mandible, while its posterior fibers contribute to retrusion of the jaw. The masseter muscle, easily palpable when clenching the teeth, provides the primary closing force and demonstrates remarkable hypertrophy in individuals who habitually grind their teeth or chew tough materials. The medial and lateral pterygoid muscles, located deep within the infratemporal fossa, provide more complex jaw movements, including protrusion, retrusion, and the side-to-side grinding motions necessary for effective food processing. The coordination between these four pairs of muscles enables the sophisticated chewing patterns that characterize human mastication and contribute to the efficient breakdown of food for digestion. The muscles of the neck demonstrate the complex balance between mobility and stability required for optimal head positioning and movement. The sternocleidomastoid muscle, one of the most prominent muscles in the neck, provides powerful flexion when both sides contract together and rotation when acting unilaterally. This muscle also assists in forced inspiration by elevating the sternum and first rib when the head is fixed, demonstrating the multiple functional roles that characterize many axial muscles. The deep neck muscles, including the longus colli, longus capitis, and the suboccipital muscles, provide fine control of head position and contribute to the maintenance of proper cervical spine alignment. These muscles work continuously during upright activities to counteract the forward bending moment created by the weight of the head and enable the precise head movements necessary for visual tracking and spatial orientation. The scalene muscles, consisting of anterior, middle, and posterior components, demonstrate the interconnection between neck and respiratory function by serving as accessory muscles of inspiration, while also contributing to lateral neck flexion and rotation. These muscles elevate the first and second ribs during forced inspiration, particularly during activities requiring increased ventilation. The muscles of the trunk represent some of the most functionally critical muscles in the human body, providing the spinal stability necessary for upright posture, while enabling the complex movements required for lifting, carrying, and manipulating objects in three-dimensional space. The concept of core stability has gained increasing recognition as fundamental to both athletic performance and injury prevention, highlighting the crucial role these muscles play in transmitting forces between the upper and lower body. The diaphragm stands as perhaps the most essential muscle in the human body, providing the primary driving force for breathing, while contributing to core stability and intra-abdominal pressure regulation. This dome-shaped muscle separates the thoracic and abdominal cavities and demonstrates remarkable structural specialization for its dual roles in respiration and postural support. During inspiration, the diaphragm contracts and flattens, increasing the vertical dimension of the thoracic cavity and creating the negative pressure necessary for air to flow into the lungs. The central tendon of the diaphragm provides a stable anchor point for contraction, while the peripheral muscle fibers insert into the lower ribs and lumbar vertebrae, creating the structural arrangement necessary for effective pressure generation. The diaphragm also contributes to core stability by increasing intra-abdominal pressure during activities requiring spinal stabilization. This pressurization effect helps support the lumbar spine and enables the transmission of forces between the upper and lower body during lifting and carrying activities. The abdominal muscles demonstrate remarkable versatility in their contributions to trunk movement, spinal stability, and respiratory function. The rectus abdominis, commonly known as the six-pack muscle, provides powerful trunk flexion while contributing to the maintenance of intra-abdominal pressure. The segmental arrangement of this muscle, created by tendinous intersections, allows for regional control and contributes to the graded nature of trunk flexion movements. The external oblique muscles, with fibers running downward and medially, provide lateral flexion and rotation of the trunk, while contributing to forced expiration through their compression of the abdominal contents. These muscles work in complex coordination patterns, with the external oblique on one side working with the internal oblique on the opposite side to produce trunk rotation. The internal oblique muscles, with fibers running upward and medially, provide complementary actions to the external obliques, while contributing uniquely to trunk stability through their deep fascial connections. The transversus abdominis, the deepest of the abdominal muscles, plays a particularly important role in core stability by creating circumferential tension around the abdominal contents and contributing to the stabilization of the lumbar spine during movement. The muscles of the back demonstrate extraordinary complexity and functional sophistication, reflecting their crucial roles in maintaining spinal alignment, enabling complex trunk movements, and providing the postural support necessary for upright activities. The erector spinae muscle group, consisting of the spinalis, longissimus, and iliocostalis muscles, provides the primary extension forces for the vertebral column, while contributing to lateral flexion and rotation. These muscles demonstrate a complex architecture with multiple fascicles spanning different numbers of vertebral levels, creating a system capable of both global spinal movements and precise segmental control. The deep intrinsic muscles of the back, including the multifidus, rotatories, and intertransversarii, provide fine control of individual vertebral segments and contribute to the maintenance of optimal spinal alignment during movement. The coordination between all axial muscles creates the complex patterns of activation necessary for maintaining upright posture against gravity, while enabling the diverse movements required for human activities. This coordination involves sophisticated neural control mechanisms that integrate information from multiple sensory systems to produce appropriate muscle activation patterns for different functional demands. 18. Major muscle groups. Appendicular muscles. The appendicular muscles represent the dynamic engines of human locomotion and manipulation, encompassing the extensive muscular systems that control the movements of the upper and lower limbs, while providing the precise control necessary for the remarkable range of human motor capabilities. These muscles demonstrate extraordinary diversity in size, architecture, and function, ranging from the massive gluteus maximus that provides propulsive force for locomotion to the tiny intrinsic muscles of the hand that enable the delicate manipulations that characterize human dexterity. The muscles of the upper limb demonstrate remarkable specialization for the complex requirements of reaching, grasping, and manipulation that distinguish human motor capabilities from those of other species. The shoulder region contains some of the most powerful muscles in the human body, reflecting the demands of supporting and moving the entire upper limb while maintaining the stability necessary for effective hand function. The deltoid muscle, forming the rounded contour of the shoulder, demonstrates a complex architecture with anterior, middle, and posterior fibers that enable different movement patterns depending on which portion is activated. The anterior fibers provide flexion and internal rotation. The middle fibers produce pure abduction, and the posterior fibers generate extension and external rotation. This functional diversity allows the deltoid to contribute to virtually every upper limb movement, while providing the primary force for arm elevation. The rotator cuff muscles represent a unique functional group that prioritizes joint stability over movement production, demonstrating how some muscles sacrifice force-generating capacity to provide the precise stabilization necessary for optimal joint function. The supraspinatus muscle initiates abduction and provides superior stabilization of the humeral head, while the infraspinatus and teres minor muscles produce external rotation and posterior stabilization. The subscapularis muscle provides internal rotation and anterior stabilization, completing the circumferential muscular cuff that maintains glenohumeral joint stability. The muscles controlling the scapula demonstrate the complex coordination required for optimal shoulder function, creating the stable yet mobile platform necessary for upper limb activities. The trapezius muscle, with its upper, middle, and lower fiber arrangements, provides elevation, retraction, and upward rotation respectively, while the serratus anterior produces protraction and upward rotation essential for overhead arm movements. The rhomboid muscles and levator scapulae provide retraction and elevation of the scapula, working in coordination with other scapular muscles to maintain optimal positioning for different arm positions and activities. The precise coordination of scapular and glenohumeral movements, known as scapulohumeral rhythm, ensures that the glenoid cavity remains optimally positioned relative to the humeral head throughout the range of arm motion. The muscles of the arm demonstrate the classic arrangement of opposing muscle groups that characterizes most limb segments, with flexors and extensors providing reciprocal control over joint movements. The biceps brachii, with its two heads arising from different locations on the scapula, provides powerful elbow flexion while contributing to forearm supination and minimal shoulder flexion. The brachialis muscle, lying deep to the biceps, provides pure elbow flexion without contribution to forearm rotation, making it particularly important for activities requiring sustained elbow flexion. The triceps brachii, with its three heads providing different mechanical advantages and stabilization roles, represents the primary elbow extensor, while contributing to shoulder extension and adduction through its long head attachment to the scapula. The coordination between these arm muscles enables the precise control of elbow position and movement necessary for optimal hand positioning during manipulation tasks. The forearm muscles demonstrate extraordinary complexity and specialization, reflecting the diverse requirements of wrist and finger movements that enable human manual dexterity. The flexor compartment of the forearm contains muscles arranged in superficial and deep layers, with the superficial muscles providing primarily wrist flexion and the deep muscles contributing to finger flexion and thumb opposition. The flexor carpi radialis and flexor carpi ulnaris provide wrist flexion, combined with radial and ulnar deviation respectively, while the palmaris longus, when present, assists with wrist flexion and tension of the palmar aponeurosis. The flexor digitorum superficialis provides flexion at the proximal interphalangeal joints of the fingers, while the flexor digitorum profundus produces flexion at the distal interphalangeal joints, creating the coordinated finger flexion necessary for grasping activities. The extensor compartment of the forearm demonstrates similar complexity, with muscles providing wrist extension, finger extension, and thumb movements that enable the release and manipulation phases of grasping activities. The extensor carpi radialis longus and brevis provide wrist extension combined with radial deviation, while the extensor carpi ulnaris produces wrist extension with ulnar deviation. The extensor digitorum provides extension of the fingers at the metacarpophalangeal joints, while the extensor indicis and extensor digiti minimi provide independent extension of the index and little fingers respectively. The muscles of the hand represent the pinnacle of muscular specialization for manipulation, with intrinsic muscles that enable the precise control necessary for fine motor tasks. The thenar muscles, controlling thumb movements, include the abductor pollicis brevis, flexor pollicis brevis, and opponens pollicis, which work together to produce the thumb opposition that is essential for human grip patterns. The hypothenar muscles, controlling little finger movements, provide similar specialized control for the ulnar side of the hand, while the interosseous muscles produce the precise finger abduction and adduction movements necessary for spreading and closing the fingers during different grip patterns. The lumbricals, arising from the tendons of the flexor digitorum profundus, produce the unique movement pattern of metacarpophalangeal joint flexion combined with interphalangeal joint extension that characterizes many fine motor activities. The muscles of the lower limb demonstrate massive size and power capabilities that reflect their primary role in supporting body weight and generating the forces necessary for locomotion. The gluteal muscles represent some of the most powerful muscles in the human body, with the gluteus maximus providing the primary propulsive force for activities such as rising from sitting, climbing stairs, and running. The gluteus maximus demonstrates remarkable architectural efficiency, with its large cross-sectional area and favorable leverage, enabling it to generate tremendous forces for hip extension and external rotation. The gluteus medius and gluteus minimus, while smaller than the maximus, play crucial roles in maintaining pelvic stability during single limb support phases of walking, preventing excessive hip drop and ensuring efficient energy transfer during locomotion. The hip flexor muscles, including the iliopsoas complex, demonstrate the importance of opposing muscle groups in maintaining joint stability and control. The psoas major and iliacus muscles, working together as the iliopsoas, provide powerful hip flexion while contributing to lumbar spine stabilization and posture control. The muscles of the thigh demonstrate the classic quadriceps-hamstring relationship that characterizes knee joint control, with the quadriceps femoris providing knee extension and the hamstrings producing knee flexion and hip extension. The quadriceps, consisting of the rectus femoris, vastus lateralis, vastus medialis, represents one of the most powerful muscle groups in the human body and is essential for activities such as rising from sitting, stair climbing, and running. The hamstring muscles, including the biceps femoris, semitendinosus, and semimembranosus, provide the eccentric control necessary for safe descent activities, while generating the hip extension forces that contribute to locomotion. The coordination between quadriceps and hamstring activation patterns is crucial for knee joint stability and injury prevention during dynamic activities. The muscles of the leg demonstrate specialization for ankle and foot control that enables the complex requirements of human bipedal locomotion. The gastrocnemius and soleus muscles, forming the calf muscle complex, provide the plantar flexion forces necessary for push-off during walking and running, while contributing to postural stability during standing activities. The anterior compartment muscles, including the tibialis anterior, extensor digitorum longus, and extensor hallucis longus, provide dorsiflexion and toe extension that are essential for ground clearance during the swing phase of walking. The lateral compartment muscles, including the peroneus longus and brevis, provide eversion and contribute to lateral ankle stability during weight-bearing activities. The intrinsic muscles of the foot, while smaller than their counterparts in the hand, play crucial roles in maintaining the arch structures that are essential for efficient bipedal locomotion, while providing the fine control necessary for balance and adaptation to irregular surfaces. The coordination between all appendicular muscles creates the complex movement patterns that characterize human locomotion and manipulation, demonstrating the remarkable integration of muscular, nervous, and skeletal systems that enables the extraordinary range of human motor capabilities. 19. Musculoskeletal integration and lever systems. The integration of the musculoskeletal system represents one of the most sophisticated examples of biological engineering, demonstrating how bones, joints, and muscles work together as coordinated mechanical systems to produce the complex movements that characterize human motor function. This integration involves not only the structural relationships between skeletal and muscular components, but also the mechanical principles that govern force transmission, leverage, and movement efficiency throughout the human body. Understanding these integrative principles provides crucial insights into how the body optimizes movement patterns while minimizing energy expenditure and maintaining structural integrity. The concept of muscle origins and insertions provides the foundation for understanding how muscular forces are transmitted to the skeletal system to produce movement. The origin of a muscle typically represents the more proximal or stable attachment point, often remaining relatively stationary during muscle contraction, while the insertion represents the more distal or mobile attachment point that moves toward the origin during contraction. However, this traditional definition can be misleading, as the relative movement between origin and insertion can reverse depending on which bone segment is stabilized during different activities. The specific locations of muscle attachments are not arbitrary but reflect millions of years of evolutionary optimization for mechanical advantage, movement efficiency, and structural stability. Muscles that require high force production typically attach close to joint centers, sacrificing speed and range of motion for mechanical advantage, while muscles that prioritize speed and range of motion attach farther from joint centers, sacrificing force production for movement velocity. The architectural features of muscle attachment sites, including tuberosities, ridges, and processes on bones, reflect the magnitude and direction of forces that muscles generate during normal activities. Larger, rougher attachment sites indicate muscles that generate substantial forces, while smaller, smoother attachment sites typically accommodate muscles that provide fine motor control or postural adjustments. The line of action of a muscle, determined by the vector from origin to insertion, defines the primary direction of force application and determines which movements the muscle can produce. Muscles with lines of action that pass anterior to a joint axis produce flexion, while those passing posterior produce extension. Similarly, muscles passing medial to a joint axis produce adduction, while those passing laterally produce abduction. Many muscles have lines of action that do not align perfectly with anatomical planes, creating moment arms for multiple movement directions simultaneously. This multiplanar capability allows single muscles to contribute to complex movement patterns and enables the nervous system to achieve efficient coordination by activating fewer muscles to produce combined movements. The moment arm of a muscle represents the perpendicular distance from the muscle's line of action to the center of joint rotation and determines the torque or rotational force that the muscle can produce about that joint. Moment arms change throughout the range of joint motion as the geometric relationships between muscle attachments and joint centers change, creating variations in muscle effectiveness at different joint positions. Understanding moment arm changes throughout joint range of motion helps explain why muscles are stronger in certain positions and weaker in others. For example, the biceps brachii has its greatest moment arm for elbow flexion at approximately 90 degrees of flexion, explaining why this position represents the strongest point in the range of motion for elbow flexion activities. The human body functions as a complex system of levers, where bones act as rigid lever arms, joints serve as fulcrums, and muscles provide the effort forces needed to overcome resistance forces such as gravity, external loads, or the weight of body segments. The mechanical advantage of these lever systems determines how effectively muscles can generate movement and overcome resistance. First-class levers have the fulcrum positioned between the effort and resistance forces, similar to a seesaw or crowbar. In the human body, the atlanto-occipital joint represents a classic first-class lever system, where the joint acts as the fulcrum. The weight of the head anterior to the joint provides the resistance, and the posterior neck muscles provide the effort force. This arrangement can favor either force or speed depending on the relative distances from the fulcrum. Second-class levers have the resistance positioned between the fulcrum and effort force, always providing mechanical advantage by allowing smaller effort forces to overcome larger resistance forces. The classic example in human movement is rising up on the toes, where the ball of the foot acts as the fulcrum. Body weight provides the resistance through the tibia, and the calf muscles provide the effort force. This arrangement explains why the calf muscles can lift the entire body weight despite their relatively modest size. Third-class levers have the effort position between the fulcrum and resistance, always favoring speed and range of motion over force advantage. The majority of muscular actions in the human body represent third-class lever systems, such as elbow flexion, where the elbow joint serves as the fulcrum. The biceps provides the effort, and any weight in the hand provides the resistance. While these systems require greater muscle force to overcome resistance, they enable rapid movements and large ranges of motion. The predominance of third-class levers in human anatomy reflects an evolutionary strategy that favors speed and versatility over pure strength. This arrangement enables the rapid, precise movements necessary for tool use, throwing, and complex manipulative tasks that distinguish human motor capabilities from those of other species. Force-velocity relationships in muscle function demonstrate how the speed of muscle contraction affects the force that can be generated. During concentric contractions, where muscles shorten while contracting. Force production decreases as the speed of contraction increases. This relationship explains why rapid movements typically cannot generate maximum force, while slow, controlled movements can produce greater force output. Eccentric contractions, where muscles lengthen while under tension, can actually generate greater forces than isometric contractions, though this type of muscle action is associated with greater risk of muscle damage and delayed onset muscle soreness. Understanding these force-velocity relationships is crucial for designing effective exercise programs and rehabilitation protocols. The length-tension relationship demonstrates that muscles generate maximum force when sarcomeres are at optimal length, where there is maximum overlap between thick and thin filaments. This optimal length typically occurs near the mid-range of joint motion for most muscles, explaining why exercises performed through full ranges of motion are generally most effective for strength development. Kinetic chains represent the concept that movement at one joint influences movement at adjacent joints, creating coordinated movement patterns that optimize efficiency and force transmission throughout the body. Open kinetic chain movements occur when the distal segment is free to move, such as during a bicep curl, while closed kinetic chain movements occur when the distal segment is fixed, such as during a push-up. Understanding kinetic chain principles helps explain how movement dysfunctions at one joint can create compensatory movements at other joints, potentially leading to injury or reduced performance. This concept is fundamental to modern rehabilitation and exercise science approaches that emphasize whole body movement patterns rather than isolated muscle actions. 20. Clinical applications and future directions. The clinical applications of musculoskeletal knowledge extend far beyond academic understanding, forming the foundation for numerous medical specialties, therapeutic interventions, and emerging technologies that directly impact human health and quality of life. From orthopedic surgery and physical therapy to sports medicine and rehabilitation engineering, the principles of musculoskeletal anatomy and physiology guide evidence-based practices that help restore function, prevent injury, and optimize human performance across the lifespan. Musculoskeletal disorders represent one of the leading causes of disability worldwide, affecting individuals across all age groups and activity levels. These conditions range from acute traumatic injuries such as fractures, dislocations, and muscle strains to chronic degenerative diseases like osteoarthritis, osteoporosis, and degenerative disc disease. Understanding the normal structure and function of musculoskeletal tissues is essential for recognizing pathological changes and implementing appropriate treatment strategies. Osteoporosis exemplifies how knowledge of bone biology translates directly into clinical practice. Understanding the role of osteoblasts, osteoclasts, and osteocytes in bone remodeling has led to the development of therapeutic interventions that can slow bone loss and reduce fracture risk. Bisphosphonates work by inhibiting osteoclast activity, while newer anabolic agents stimulate osteoblast function to promote new bone formation. Exercise prescriptions based on bone loading principles help optimize bone density throughout life. Arthritis, particularly osteoarthritis, represents another area where fundamental knowledge of joint structure and function informs clinical management. Understanding the role of articular cartilage, synovial fluid, and subchondral bone in joint health has led to treatments ranging from viscosupplementation with hyaluronic acid to joint replacement surgery. The recognition that mechanical factors play crucial roles in arthritis development has emphasized the importance of maintaining proper joint alignment and muscle strength. Sports medicine represents a field where musculoskeletal knowledge is applied to optimize athletic performance, while minimizing injury risk. Understanding muscle fiber types helps explain individual differences in athletic capabilities and guides training program design. Knowledge of biomechanical principles informs technique analysis and equipment design, while understanding tissue healing processes guides return-to-play protocols following injury. The prevention and treatment of overuse injuries relies heavily on understanding how tissues adapt to mechanical stress and the concept of tissue tolerance versus tissue demand. When demands consistently exceed tolerance, overuse injuries develop. This principle guides load management strategies that progressively increase training demands while allowing adequate recovery for tissue adaptation. Physical therapy and rehabilitation represent clinical applications that rely extensively on musculoskeletal knowledge to restore function following injury or surgery. Understanding normal movement patterns enables therapists to identify movement dysfunctions and design targeted interventions. Knowledge of muscle actions and biomechanical principles guides exercise selection and progression, while understanding tissue healing timelines informs treatment planning. The concept of motor learning and neuroplasticity has revolutionized rehabilitation approaches, recognizing that the nervous system's ability to adapt and reorganize extends well into adulthood. This understanding has led to task-specific training approaches that emphasize functional movements rather than isolated muscle strengthening, improving outcomes for patients with neurological and musculoskeletal conditions. Ergonomics represents another clinical application that uses musculoskeletal principles to prevent work-related injuries and optimize human performance in occupational settings. Understanding joint mechanics and muscle function guides workstation design, tool development, and work practice recommendations that minimize stress on the musculoskeletal system while maintaining productivity. The aging process creates unique challenges for the musculoskeletal system, with age-related changes including decreased bone density, reduced muscle mass and strength, sarcopenia, decreased joint mobility, and altered balance and coordination. Understanding these changes has led to targeted interventions such as resistance training programs that can slow or reverse age-related muscle loss, and balance training that reduces fall risk. Emerging technologies continue to expand the clinical applications of musculoskeletal knowledge. Computer-assisted surgery uses detailed anatomical knowledge combined with imaging technology to improve surgical precision and outcomes. Robotic systems for rehabilitation leverage understanding of normal movement patterns to provide precise, reproducible therapy interventions. Tissue engineering represents a promising frontier where understanding of cellular biology and biomechanics is being applied to develop replacement tissues for damaged musculoskeletal structures. Researchers are working to create artificial cartilage, engineered bone grafts, and even whole joint replacements that can integrate with and function like natural tissues. Regenerative medicine approaches, including stem cell therapy and platelet-rich plasma treatments, are based on understanding how the body's natural healing mechanisms work and how they can be enhanced or directed to improve outcomes. While still in early stages, these approaches hold promise for treating conditions that have traditionally been difficult to manage. Biomechanical modeling and simulation technologies allow researchers and clinicians to predict how interventions might affect musculoskeletal function before implementing them in patients. These tools can help optimize surgical techniques, design better prosthetic devices, and predict long-term outcomes of various treatment approaches. Wearable technology and motion analysis systems are making it possible to monitor musculoskeletal function in real-world settings, providing insights into how interventions perform outside controlled clinical environments. This technology enables personalized treatment approaches based on individual movement patterns and activity levels. Gene therapy and molecular medicine represent future directions that may allow direct modification of cellular processes involved in musculoskeletal health and disease. Understanding the genetic basis of conditions like muscular dystrophy and osteogenesis imperfecta may lead to treatments that address these conditions at their molecular roots. The integration of artificial intelligence and machine learning with musculoskeletal knowledge is beginning to enable more sophisticated analysis of movement patterns, prediction of injury risk, and optimization of treatment protocols. These technologies may eventually enable truly personalized medicine approaches that account for individual, anatomical, physiological, and genetic variations. Prevention strategies based on musculoskeletal science continue to evolve, with increasing emphasis on identifying and addressing risk factors before problems develop. This includes screening programs for osteoporosis, movement analysis to identify injury risk patterns, and educational programs that teach proper body mechanics and exercise techniques. The future of musculoskeletal medicine will likely involve increasingly sophisticated understanding of the molecular mechanisms underlying tissue function, more precise diagnostic capabilities, and treatment approaches that can be tailored to individual patient characteristics. As our knowledge continues to expand, the potential for improving human mobility, reducing pain and disability, and enhancing quality of life through musculoskeletal interventions will continue to grow. The remarkable complexity and adaptability of the musculoskeletal system ensure that there will always be new discoveries to be made and new applications to be developed. This system, which enables everything from the most delicate manual tasks to the most powerful athletic performances, represents one of the most fascinating and practically important areas of human biology, with direct relevance to virtually every aspect of human health and performance.